disturbance during the sessions. Thank you so much. A very warm welcome to all our guests here. Thank you so much for your eminent presence here today. It is my honor and delight. I feel really privileged to welcome one and all our guests, esteemed chairpersons, speakers, delegates, senior international colleagues, and our young investigators. It gives us immense pleasure to welcome you at the 13th India Probiotic Symposium Day 2. The theme of the symposium this year is Science-Based Evidence on the Benefits of Probiotics for Human Health. For the first session of the day here, I would request the humble presence of Professor A.K. Srivastava and Dr. Saurabh Nagpal to chair the session. Can we give a round of applause to the dignitaries here? I would like to give a brief introduction about our esteemed chairpersons here, starting with Professor A.K. Srivastava. Professor Dr. A.K. Srivastava is presently Vice Chancellor of DU VASU Mathura. He's a distinguished dairy and animal scientist. He's the Chairman of Agricultural Scientist Recruitment Board, Ministry of Agriculture and Farmers Welfare, Government of India. He has also served as Director and Vice Chancellor of ICAR National Dairy Research Institute, Karnal. He has been decorated with numerous prestigious awards and honors, including some most important: ICAR Jawaharlal Nehru Award, International NOCIL Award, National Alarson Awards, and last but not the least, National Education Awards, and many, many more. Can we have a round of applause for him, please? Thank you so much. Next, we have Dr. Saurabh Nagpal, who has done his master's in prosthodontics and implantology after completing his bachelor's from Government Dental College, Patiala. He's director of Matrix Dental and Skin Lounge, Vasant Vihar, which has been adjudged as the best dental clinic in Delhi NCR by reputed agencies like Times of India, NDTV, Assocham, etc. Matrix is currently serving 50 plus embassies and overseas missions of various countries placed in India and is one of the favorite destinations for expats seeking dental treatment. Matrix has been pioneering field of dental tourism for almost 2 whole decades. Practor.com consistently ranks Dr. Saurabh Nagpal at top rank in Delhi since last 6 to 7 years. His favorite quote, I must say, in cricket and in dentistry is, India is not less than any other country. So I would now be requesting our honorable chairpersons to kindly take a lead and start the first session of the day. Thank you so much.
Thank you, ma'am. Thank you very much. Well, friends, I, on behalf of my colleague, Dr. Saurabh Nagpal, extend heartiest welcome to all the participants, delegates, and students who are today in this hall to attend a very important seminar, and it is very important session. In this session, me and Dr. Saurabh Nagpal will be chairing the session, and we are having 6 speakers. Out of these 6 speakers, one is keynote speaker, Dr. Michael T. Bailey, and 5 other speakers. So keynote speaker is having 30 minutes and other speakers are having 20 minutes each. I will request all the speakers to kindly stick to the time. 2 minutes before we will give some indication, and then we will request to wind up in time so that we can finish this technical session in time. With this 6 speakers, let us start our technical session. First, I will request Dr. Michael T. Bailey. Dr. Michael T. Bailey, he is Associate Professor of Pediatrics in Ohio State University, Medical School, Columbus, Ohio, USA. Dr. Michael, is he here?
I'm here, yes. I'm here. Can you hear me?
Okay, Michael. Give me 1 minute. Let me introduce you.
Sure.
So Dr. Michael Bailey is an associate professor of pediatrics at the Ohio State University College of Medicine and a principal investigator in the Center of Microbial Pathogenesis at the Research Institute at Nationwide Children's Hospital. He has conducted pioneering studies showing that the body's physiological response to stress changes the composition of the gut microbiota, which in turn impact immune system functioning and behavioral responses. He is also focused on identifying mechanism by which gut microbes interact with their host with the goal of developing microbes-derived treatment for inflammatory and behavioral diseases. He has published more than 100 manuscripts and books chapter on various aspects of the multi-directional interaction between gut microbes, immune and nervous system functioning, disease susceptibility, and behavioral response. With this brief introduction, I welcome you, Dr. Michael, and request you to start your presentation. I request that you are having 30 minutes with you, so finish in time in 30 minutes. Okay,
Thank you.
please, time is yours now.
Thank you. Hang on just a second here. Great. Thank you for the introduction and thanks for the invitation to participate today. I'm very happy to be sharing some of our data with you and sharing some of our thoughts with you this morning. So just for disclosure, I do have an interest in biosciences, and I will talk about one of our projects later in the presentation. So I've had a longstanding interest in trying to understand the impact that the body's physiological response to stress has on susceptibility and resistance to different types of diseases. And of course, we know that when the body is exposed to stress, and this could be physiological stress, it could be psychological stress, or even social stress, a number of different pathways within the body become activated. And two of these pathways that are well conserved across different types of stressors are the sympathetic branch of the autonomic nervous system, as well as activation of the HPA axis. And these pathways lead to the production of glucocorticoid and catecholamine hormones and neurotransmitters. And of course, we're used to thinking about these hormones and neurotransmitters for their effects on the physiological stress response itself. They're responsible for the classic fight or flight response, but we also know that these hormones and neurotransmitters have a strong effect on the gastrointestinal tract. And in fact, we know that gastrointestinal physiological functions such as the secretion of gastric acid, motility patterns in the intestine, the secretion of bile and mucus, all of these can be significantly changed during a stress response. We also know that the stress response can significantly alter immune system activity, including components of the immune system that can impact the microbiota, such as secretory IgA and the secretion of antimicrobial peptides, also can be changed during a stress response. And of course, there's a lot of interest right now in the fact that bacteria can both respond to and produce compounds that are traditionally used as hormones and neurotransmitters by their mammalian hosts. So early in our studies, this led us to a very simple question of whether exposure to stress can change the composition of the gut microbiota. And so in my laboratory, we typically use two different murine stressors to test our hypotheses. One of these stressors is a model of repeated social defeat that we call social disruption, whereas the other model is a prolonged restraint stressor. And these stressors may seem sort of random or artificial, but they're actually very well validated stressors that are widely used in the behavioral and biomedical sciences. And they're widely used because these stressors will reliably activate both the HPA axis and the sympathetic nervous system. These stressors also will lead to stress indicative behaviors like anxiety-like behavior and depressive-like behavior. And both stressors will also have an impact on immune system functioning. So in our earliest studies, we exposed mice to either a social disruption stressor or the restraint stressor. And at the end of the stressor, we removed the colon and used 16S sequencing to test whether exposure to these stressors would change the composition of the colonic microbiota. And what you can see is that regardless of whether mice are exposed to the social disruption stressor or to the prolonged restraint stressor, exposure to either of these stressors lead to significant changes in microbial composition in the colon. And these early observations were very instrumental in developing this idea of a bidirectional communication between the nervous system and the gut microbiota through the traditional brain-gut microbiota axis, with our studies really focusing on sort of top-down communication, just illustrating that activation of the nervous system, such as through stress, or different conditions that are associated with nervous system activities, such as anxiety and depression, lead to changes in the composition of the gut microbiota. Other labs have also used fecal transplants to show that these changes in the microbiota are physiologically important. So for example, you can take the microbiota from a patient with anxiety or depression, transplant that into a germ-free mouse, and show that these changes in the microbiota lead to significant effects on the nervous system and behavioral responses in the recipient mice. So truly, this is a bidirectional interaction between the nervous system and the gut microbiota. And now that this is fairly well established in the literature, our challenge is really trying to understand the pathways through which these interactions occur. So of course, we know that the vagus nerve is a very important component of the brain-gut microbiota interactions. We also know that there are a number of metabolites that can be both produced and recognized by both the host and its microbiota. And we also know that the immune system is often a mediator between interactions between the microbiota and the nervous system. What really has not been very well studied, however, is whether the intestinal epithelium also plays a role in the brain-gut microbiota axis. And of course, we know that the epithelium is very heavily innervated by the nervous system. And the epithelium also contains receptors and transporters for a number of different microbial compounds that then can be either transferred, or a signal can be transmitted across this epithelial barrier to stimulate the enteric nervous system. But of course, there are many barriers that prevent these bacteria and bacterial products from just simply diffusing across the epithelial barrier and stimulating the enteric nervous system, with one of those barriers being the mucus layer. There's this well-defined mucus layer in the intestine that we know separates bacteria in the lumen of the intestine from the epithelial layer. So we wanted to know whether this mucus barrier is an important component of the brain-gut microbiota axis. And to begin to test this, we've focused on our social defeat stressor that we know leads to a low-grade inflammatory response and also leads to a variety of different behavioral changes, including differences in working memory, which is what we focused on in this study. So you can see that in mice that are exposed to the stressor and then tested on the Y maze to test for spatial working memory, animals that are exposed to stress have a significant reduction in working memory on the Y maze. And as I alluded to, we know that exposure to the stressor leads to a low-grade inflammatory response. So we measured a number of different inflammatory mediators to determine whether any of these were related to behavior on the Y maze. And there really was only one inflammatory mediator that seemed to be related to this working memory task, and that was levels of LPS binding protein in the serum. In general, animals that had higher levels of LPS binding protein, which was significantly increased in our stressed animals, also had the lowest amounts of working memory on the Y maze. So this was important to us because we know that LPS binding protein is an acute phase protein. It's produced by the liver during an infection or during severe inflammatory reactions. But we also know that LPS binding protein can be produced by the intestinal epithelium. And it's produced by the epithelium when bacteria are actually able to penetrate the mucus layer to stimulate the epithelial cells to produce LPS binding protein. So we wanted to know whether our exposure to our stressor was associated with disruption in this mucus layer, allowing bacteria to come into contact with the epithelium. And we chose to test this using fluorescent in situ hybridization to stain our bacteria in the lumen of the intestine, and couple that with a lectin-based stain to stain our mucus layer, and then a DAPI counter stain for our epithelial cells. And what you can see is in control animals, there is this very well-defined mucus layer in the colon that spatially separates bacteria from the epithelial cells. But in our animals that are exposed to stress, we see a significant depletion of this mucus layer. And in some cases, it actually looks like these bacteria are coming into contact with our epithelial cells. In addition, we characterized the bacteria in the colon using 16S sequencing. And we found that there were a couple of taxa whose relative abundances were significantly increased in stressor-exposed animals, particularly bacteria in the genus Myribaculum, which is potentially important because we know that Myribaculum has been associated with low-grade inflammatory responses in other models. In addition to these increases in Myribaculum, we also observed a significant reduction in bacteria in the genus Akkermansia. And this makes sense to us because Akkermansia are known to use mucus as a carbon source. So the reduction in Akkermansia is very consistent with the reduction in mucus and the reduction in available carbon for available nutrients for the Akkermansia. So, we, had evidence that these bacteria were potentially coming into contact with the intestinal epithelium. We observed higher levels of LPS binding protein in circulation, and we wanted to know whether the stress-induced increase in LPS binding protein really was due to differences in the microbiota, or at least involved the microbiota. And so we exposed mice to our stressor, and we treated those animals with an antibiotic cocktail to try to reduce the microbiota. And so in vehicle-treated animals, you see the stress-induced increase in LPS binding protein in the serum. But if we treat the animals with our antibiotic cocktail, we no longer see a stress-induced increase in LPS binding protein. And to go along with that, when we test their behavior on the Y maze, we no longer see the stress-induced reduction in working memory if the animals are treated with antibiotics during exposure to our stressor. So in addition to measuring their behavior, we also wanted to assess the brain for different neural correlates of these behaviors. And we specifically focused on neuronal FOSB staining in the hippocampus, as well as microglial IBA1 staining also in the hippocampus. And what you can see is that in stressor-exposed animals, there's a significant increase in phosph-B staining in the hippocampus, but this is only in animals that are exposed to stress and the vehicle. If instead those animals are treated with antibiotics, you no longer see the stress-induced increase in phosph-B staining. And similarly, when we look at the microglia, there's a significant increase in microglial IBA1 staining in the hippocampus, but that's only in animals treated with the vehicle. If we instead treat with antibiotics, we no longer see the stress-induced increase in IBA1 staining. So we wanted to determine whether this LPS binding protein could potentially be the mediator between the microbiota and these differences in behavior that we are observing. And so LPS binding protein, as the name implies, binds to LPS. And the LPS and the LBP complex then binds to CD14 on immune cells that helps to activate toll-like receptor 4, leading to the induction of an inflammatory response. So we use CD14 knockout mice to test the importance of this pathway for inducing this inflammation and these behavioral responses. And what you can see is that CD14 knockout mice that are exposed to the stress do not show a stress-induced reduction in spatial working memory on the Y maze. And to go along with that, these CD14 knockout mice that are exposed to stress do not show a stress-induced increase in microglial staining or a stress-induced increase in neuronal phosph-B staining. And so what we think this means is that during a stress response, the stress response leads to a reduction in the mucus layer in the intestine, allowing bacteria to come into contact with the intestinal epithelium. That bacterial contact with the epithelium leads to an increase in protective responses like LPS binding protein that goes on to trigger this low-grade inflammatory response. This inflammation can feed back to the intestine, further leading to further dysregulation of homeostasis in the intestine. But that inflammation can also affect the brain, leading to changes in behavior such as anxiety and depression, or in the case of this study, differences in cognitive behavior. Interestingly, in our human studies where we have looked in the blood of human patients with depression or anxiety, or even human participants that are exposed to a stressor like marital conflict, we also see significant increases in LPS binding protein in the serum, as well as inflammatory cytokines. And we're very interested in whether these inflammatory markers may be related to differences in behavior, including differences in cognition during these different periods. So the fact that we can observe that the microbiota are involved with neuroinflammation and behaviors that may be driven by neuroinflammation has led us to begin testing whether we can target the microbiota to try to improve neuroinflammation and related behaviors. And of course, the methods that we're using to target the microbiota are probiotics and also prebiotics. And interestingly, when we started using probiotics, and the probiotic that we focus on is Lactobacillus reuteri, or Limicil Lactobacillus reuteri, as it's recently been renamed. We found that when we administered Lactobacillus reuteri in just a normal traditional planktonic state, the probiotic really had very limited effects on inflammation. It could reduce inflammation, but only if that inflammation was sort of in a mild state. Once that inflammation became more severe, the bacteria were relatively ineffective at reducing inflammation. So a group of us at Nationwide Children's Hospital began to determine whether there were different strategies we could use to increase the efficacy of our probiotic. And the strategy that we came up with was to adhere the bacteria to the surface of a microsphere. And the microsphere that we're using is just the dextronomer microsphere, to allow those bacteria to adhere to the microsphere and to begin to create a biofilm on the surface of that microsphere. Now, what's interesting about these microspheres is that they're porous, so we can load these with different nutrients that we know the bacteria will use to create a more robust biofilm. So for example, we can load sucrose into these microspheres. The sucrose slowly diffuses out of the microsphere, and the Lactobacillus reuteri on the surface of the microsphere will utilize the sucrose to create a biofilm, which is colored yellow on this electron micrograph. And again, this is a strategy that was co-developed with Steve Goodman, Gail Besner, Lauren Bakelitz, and I at Children's Hospital, and has been licensed to Scioto Biosciences to further develop. So interestingly, this strategy to increase the biofilm on the surface of these microspheres significantly increases the ability of our probiotic to reduce inflammatory responses, even very severe inflammatory responses, such as inflammation that occurs with necrotizing enterocolitis in newborn infants is significantly reduced using this strategy. And we've shown this in a rat model as well as a mouse model, and more recently in a pig model as well. We also can prevent severe bacterial infections in the intestines, such as Clostridioides difficile infection. More recently, we've become interested in a recent finding where we've shown that this probiotic strategy also enhances neurodevelopment. And we've first shown this in rat pups that have survived necrotizing enterocolitis. And we're now shifting our focus to autism spectrum disorder. And in fact, our startup company, Scioto Biosciences, has just completed a phase 1 clinical trial using a clinical trial in participants with autism spectrum disorder. Of course, it's a phase 1 trial, so they were testing the safety of our strategy, which was found to be safe. But some of our secondary outcomes in our autistic participants makes it seem like this may be a very important strategy to improve some of the symptoms associated with autism spectrum disorder. So we're very interested and very hopeful for our phase 2 clinical trials that should be starting later this year. But in addition to using probiotics, we've also been using prebiotics to determine whether prebiotics can improve inflammatory responses to affect the brain and behavior. And so I'm just going to talk about one of our studies where we've used the prebiotic galacto-oligosaccharide. And so mice were fed a diet containing GOS, or control diet, for 3 weeks. And then we assessed the brain and behavior. We assessed several different cytokines and chemokines in the brain, and we're assessing open field behavior for behavior in the open field for assessment of anxiety-like behaviors. We're also assessing the metabolome in both the intestine and the serum, and the microbiome in the intestine. And so, as I mentioned, we're focused on behavior in the open field. And what you can see is that mice that are fed the prebiotic diet spend more time in the center of the open field. And this is generally interpreted to mean that these animals have a significant reduction in anxiety-like behavior. So in addition to this reduction in anxiety-like behavior, these animals also have a reduction in a few cytokines and chemokines in the brain. Not all of them are reduced, but there are a few chemokines and cytokines that are reduced. Specifically, CCL2 is significantly reduced in the prefrontal cortex, as well as TNF-alpha is being reduced in the prefrontal cortex, with a trend for CCL2 also being reduced in the hippocampus. And what's interesting is that gene expression for these chemokines and cytokines seems to be significantly correlated with behavior in the open field. So in general, animals that had the lowest levels of CCL2 and TNF-alpha had the lowest amounts of anxiety-like behavior. So then, of course, the question is how? How is it possible that a prebiotic that is used by gut microbes can impact the brain and behavior? And we began to focus on metabolites that may be mediating this response. So to begin to look at the metabolites, we fed mice our diet and then collected the colon, sent the contents of the colon to a company called Metabolon to assess the metabolome, and found that there were a number of metabolites that were either significantly increased or significantly decreased when these animals were fed the prebiotic diet. But interestingly, none of these metabolites were significantly correlated with behavior. So that led us to also assess the serum metabolome. And again, we found a number of metabolites that were either increased or decreased in the serum. But here there were a handful of metabolites that were significantly correlated with behavior in the open field. And one of those metabolites that correlated with behavior was methyl-indole-3 acetic acid, or MI3A. And you can see that MI3A levels in the serum were correlated with time in the center of the open field in both animals that were fed the control diet or fed the prebiotic alone. So in general, higher levels of MI3A was associated with lower levels of anxiety-like behavior. Interestingly, though, MI3A levels in the serum were not quite statistically significantly increased in animals fed our prebiotic. So that led us to also look for related compounds that may be affected by the prebiotic. And as we suspected, one of those compounds, indole-3 acetic acid, was significantly increased by the prebiotic. Interestingly, though, I3A was not correlated with behavior. Only the MI3A was correlated with behavior. Regardless, I3A caught our attention because indole-3 acetic acid is a well-known microbial metabolite of tryptophan. And of course, there's a lot of interest right now in the ability of bacteria to metabolize tryptophan and produce a number of different metabolites that can affect the immune system and also affect behavioral responses. So we assessed the microbiome, again, using 16S sequencing, and we found that there were 2 bacteria whose relative abundances were significantly increased by the prebiotic. One of them was an unclassified genus of Lactospiraceae, and the other was Akkermansia. There's a significant increase in Akkermansia in animals fed the prebiotic diet. And this is interesting to us because we know that the primary genus within the the primary species within the genus Akkermansia, which is Akkermansia muciniphila, contains genes necessary for metabolizing GOS. So that's why we have this expansion in Akkermansia. But these bacteria also contain genes necessary for metabolism of tryptophan. And so we think what happens is that when we feed the prebiotic, we have an expansion of these Akkermansia that are able to metabolize dietary tryptophan, leading to an increase in tryptophan metabolites like the indole-3 acetic acid. And we know that these indoles are widely methylated within the liver. They might also be methylated within the intestine, but definitely within the liver, leading to the production of methyl indole-3 acetic acid. And so we wanted to know whether methyl indole-3 acetic acid could be directly contributing to changes in behavior. And so we did an experiment where we simply injected methyl indole-3 acetic acid into our animals, and 2 hours later measured behavior in the open field and found that injection of MI3A significantly reduced anxiety-like behavior in the open field. Interestingly, we observed that with methyl I3A, but when we injected I3A and measured behavior 2 hours later, we did not see a significant decrease in anxiety-like behavior, only if we injected the MI3A. So we also measured gene expression in the brains of these animals that were injected with MI3A and found that CCL2 was reduced in both the prefrontal cortex and the hippocampus. There are also trends for TNF-alpha to be reduced in both the prefrontal cortex and the hippocampus, but there was no effect for IL-6. So next, we wanted to know whether it was possible that this MI3A has a direct effect on microglia, which are the primary cytokine-producing cells within the brain. And so we cultured microglia in vitro. We're using a cell line in this experiment, the BV2 microglial cells, stimulated those cells with LPS, as well as with different concentrations of either MI3A or I3A. And what you can see is that when we stimulate these cells in the presence of MI3A, the MI3A significantly reduces CCL2 levels in the supernatants of these cells. But interestingly, I3A had no effect on CCL2. I will point out, though, that we see similar effects with TNF-alpha, with MI3A reducing TNF-alpha and I3A having no effect on TNF-alpha. But when we measure IL-6, the pattern is reversed. IL-6 is reduced by IL-3A. IL-6 is reduced by I3A, but MI3A does not reduce IL-6. And we're not sure yet why we are observing those differences and what those differences mean for behavior. But certainly for CCL2, we see this reduction in cells that are treated with MI3A. And this is very consistent with our in vivo data, where, again, what we think happens is that when we feed our prebiotic, we have an expansion of Akkermansia that are able to metabolize dietary tryptophan, leading to indole-3 acetic acid. The indole-3 acetic acid is methylated by the host, and we think it's this methylated form that affects behavior because of the effects of MI3A on chemokines like CCL2 or cytokines like TNF-alpha that are produced by our microglial cells. So just to wrap everything up, our data are really leading to this idea that the intestinal barrier plays a very key role in the brain-gut axis, and that actually the mucus layer might be an important part in the brain-gut axis that to date really has not been very well studied, and it may play a very important role in regulating inflammatory responses. We have also shown that microbe-directed neuroinflammatory responses directly contribute to behavioral outcomes. We've shown this with spatial working memory, and we have ongoing studies where we are looking at both anxiety-like behavior and depressive-like behaviors in our mouse models. And finally, we're very interested in whether we can sort of tune microbial metabolites to reduce neuroinflammatory responses and use these microbial metabolites as a therapeutic to reduce inflammation and improve behavioral outcomes. So I'd just like to wrap up by thanking the members of my lab, as well as my collaborators, both at Ohio State and Nationwide Children's Hospital, and of course, our sources of funding. And I will stop there and take any questions. Thank you.
Thank you very much, Dr. Michael Mele. Thank you very much. Please stay with us for a couple of minutes. If there is any question, you can respond to them. Now this paper is open for question. Yes, Dr. Ganguly, please. Mike.
Yes.
Yes, there is a question for you. Please just listen and then respond. Yeah.
So,
Okay.
so one thing we get, Dr. Bailey, that avoid aggressive people around you because your aggressive mice gave. But my question is twofold. That
Mhm.
I find that although we got a paper on Parkinson's yesterday, but we find that in the ataxic children, this could be of great value. But what I see here that I3A could be better than MI3A because I3A ultimately leads into metabolic pathways which are ultimately beneficial because it moves NF kappa B towards a better behavior. The one thing which I didn't understand that making biofilms, could it be because biofilms ultimately we want to break because what happens in the biofilm that two populations are generated. The one population is slowly multiplying, another population is multiplying normally, and the quorum sensing molecules creates the biofilm. So, and antimicrobial resistance also develops in those situations. So, this is it good to name it as a biofilm, or we are enhancing the addition of these and around cytodex kind of a molecule so that they can create a coat and are better able to communicate within the system. So I was just and was the LPS binding protein a good marker to measure stress? Did you measure catecholamines also to see that whether there is any correlation with these? Like do catecholamine decrease after intervention?
Right. Great question. So I'll start with the first one, the biofilm. So, we're calling it a biofilm just based on the presence of the extracellular matrix. So, we can observe extracellular matrix there. So we do think that these bacteria are forming a biofilm. And in this form, these bacteria, at least in vitro, are more resistant to acids. So they're very resistant down to a pH of 2, which is consistent with the biofilm as well. The biofilm in general is more resistant to host defenses. And in some of our ex vivo studies, the bacteria on the biofilm seem to be better able to adhere to intestinal mucus and also adhere to intestinal epithelial cells. So having said all that, we don't really know why exactly this formulation is so effective in vivo. We do know that the bacteria persist in vivo for a longer period of time. So it may just be that we are able to have higher numbers of viable bacteria for longer periods of time, or it could be that these bacteria are producing different metabolites, including quorum sensing molecules, because of this biofilm state. So those are open questions that we just don't know at this time. So the other question of whether we measured catecholamines in our studies where we measured LPS binding protein, and to be honest, the answer is no. So we've measured catecholamines in these models previously. So we know that the catecholamines are increased, and they're increased along with our inflammation. So they both tend to parallel each other. But I don't know really if the catecholamines and the LPS binding protein correlate with each other. And if we try to treat our animals to reduce LPS binding protein, if we also reduce catecholamines, that's a good question. So, thanks.
Can I? Thank you, Dr. Michael. Thank you.
Yeah, I have one question. This is Dr. Shastri.
Please be loud, please.
Yeah, Dr. Bailey, it was excellent presentation and really nice work. I enjoyed.
Thank you.
I had a query, like, is the stress-induced neuroinflammation, is it physiological or pathological? Should we be targeting it? Because when you are exposed to stress, is this a physiological response of the body? Then should we be changing it?
Yeah, that's a great question. So, yeah, I would say it's a physiological response. So it is a natural response. I think where it could potentially become pathological is we know that in these animal models, it's sometimes very prolonged. So we exposed our animals to a stressor for we do it for 6 days, but we can detect anxiety for 3 weeks later. And it's probably that longer period that is more relevant than the acute period, if that makes sense. So the acute phase is really physiological. The more prolonged period would be more pathological. Right now in our studies where we're assessing the neuroinflammation, we're really only looking at that acute phase. And so that's a good point that it would be interesting to look at the prolonged pathological inflammation.
Thank you. Yes, last question. Yeah, please. Mike, you can have.
Professor Bailey, that was real good work. And I'm a pediatrician. I would like to ask you, did you use Uteri for infantile colics? Because you worked in the neonatal setup as well. And obesity, which is going to be a huge problem by 2030. And you mentioned autism. These are the problems we are facing in India as well.
Yes, so we haven't used Reuteri yet in the NICU. We are still working with the FDA to get our phase 1 clinical trial approved in neonates, but we're very hopeful. So far, everything has been done in animal models with our, with our newborn models. And so, yes, autism spectrum disorder, there have been a couple of other groups that have shown that Lactobacillus reuteri can increase oxytocin levels and seems to improve animal models of autism. In fact, in our studies, when we use our probiotic platform, we can see a further increase in oxytocin. So, that's the idea that this may be how we are going to improve autism spectrum disorder. So that's where the hope is. And then I think your other question was obesity, and we have not done anything with obesity just yet, but that's an interesting question.
Thank you.
Thank you, Michael. Last question from chair, Dr. Saurabh.
So I just want to ask you one thing. What you explained chemically here is what we have seen in our practice clinically. So there are patients arriving with stress ulcers, and I've seen most of dentists not being able to diagnose it. So what I have seen is by the time first doctor, second doctor, third doctor patient is running on to, it gets resolved. So I want to understand, is it self-limiting? Over time, it will resolve on its own? Or is it that multiple doctors kept on prescribing to somebody, some went with antivirals, some went with probiotics. Patient also must have changed diet, must have tried to acclimatize with the anxiety or whatever the scenario, body acclimatizes over time. So is it self-limiting or it will dictate the body?
Is the disruption in the microbiota self-limiting?
Yeah.
Is that the question?
Yeah.
Yes. I mean, if there are no other insults, I think it will eventually resolve. In our animal models, we know that, the microbiota is disrupted with stress very, very quickly, actually, but then will return closer to normal within 24 hours, and then normal in 3 to 7 days.
We saw same thing in Corona as well. So patients with the altered microbiota were arriving, and probably that is also one reason, apart from other reasons, that the taste sensation, smell sensation were getting lost. So
Right.
again, that is again one place where we saw it was self-limiting.
Yes. Yep. Yes. So I think it is, again, as long as there's no further stimulus there. I think what often happens is you get this disruption in the microbiota and then also get an inflammatory response to the dysbiosis. And that inflammatory response then feeds back and causes further dysbiosis. And so you have this loop. But I think as long as you don't trigger this inflammatory response, you will see a transient change in the microbiota, but it will reestablish a baseline fairly rapidly.
Why I'm asking you is I'm also one of the patients suffering with this. So there were years altogether. Every year I used to get stress ulcers. I tried all the medications, everything. And then later on, I realized it is related to my traveling as well as dietary habits. Whenever it is excessive traveling along with dietary habits are there, so it gets exaggerated. Eventually, I realized that it is self-limiting as well, but it takes good 1 week, 10 days to settle down.
Mm-hmm. Yeah, interesting. So yeah, I think that makes sense with what we see in the animal models as well.
Thank you. Thank you. Michael Bailey, thank you very much. And for a wonderful presentation, science-based evidences for all these things. Let us give a big clap to him. It is midnight, I think, in Ohio.
It is 1:30 a.m.
Oh, now please go in bed and have good sleep.
Thank you.
Thank you. Thank you very much.
Thank you.
Thank you. Okay. Okay. Good night. Good night. Thank you. Okay, friends, after wonderful presentation by Dr. Michael T. Bailey, now we are coming to the second speaker, Mr. Takua Sugimoto. Yes, Mr. Sugimoto, please come. Takua Sugimoto is currently the Associate Senior Researcher at the Analytical Science Department of the Yakult Central Institute. He has been involved in research in the field of the intestinal microbiota, particularly on the relationship between gut microbes and infectious complications from surgery or adverse events of cancer chemotherapy, and the use of symbiotic intervention to minimize them. He has published more than 10 original papers in peer-reviewed international journals. His main interest is to contribute to develop new measures, especially probiotics, for the medical field to establish the therapeutic effects and improvement of QOL.
Thank you.
Mr. Sugimoto, now floor is yours. 20 minutes is for you for presentation, and then we will have 4 or 5 minutes, some questions if they are there. Okay, please.
Thank you for your kind introduction. Hello, everyone. My name is Takuya Sugimoto, and I'm from Yakult Central Institute, Japan. Before starting my presentation, I would like to thank the organizers of Yakult Probiotic Symposium for giving me the opportunity to speak. Today, I would like to talk to you about prevention of postoperative infectious complications with symbiotics. Usually, the gut microbiota is well balanced with high level of beneficial or commensal bacteria and low level of harmful or opportunistic bacteria. However, the gut microbiota is disrupted by aging process and certain lifestyle habitats. This condition is known as dysbiosis. Dysbiosis causes negative phenomena such as increased gut permeability or decreased level of beneficial active molecules. It has been recently reported that the dysbiosis can result from high invasive surgery, cancer chemotherapy, or systemic inflammation, and that dysbiosis is associated with infectious complications. So is there a way to improve dysbiosis? It has been recently reported that diet and lifestyle modification, as well as as well as the intake of prebiotics or probiotics improve dysbiosis. Therefore, we hypothesize that the administration of symbiotics, the combination of prebiotics and probiotics, can reduce the infectious complication by further improving gut dysbiosis. This slide shows the list of randomized control trials that examine the use of symbiotics for patients undergoing abdominal elective surgery. Various combinations of prebiotics and probiotics were used in various surgical cases. The table is sorted by the symbiotics used. Here, the upper part of this table is use our symbiotics in Japan, but however, the various symbiotics is various combinations of but other studies are being conducted around the world in various surgical cases. So the administration of symbiotics is a very promising method for preventing infectious complications. The next slide is, I will discuss our symbiotic in detail. Our symbiotics is the combination of probiotics, the lactic acid producing bacteria, Lactobacillus parasiticida, and acetic acid producing bacterial strain Yakult, and prebiotics in the form of oligosaccharides, which are said to be digested by bacteria. We examine whether our symbiotics is effective in suppressing post-operative infectious complications in patients scheduled to undergo major hepatectomy. Here, we saw overall incidence of post-operative infectious complication is significantly lower in the symbiotics group than the control group. Furthermore, we show the second trial that post-operative plus pre-operative administration of symbiotics further reduce the incidence of infectious complications. These results indicated that our symbiotics is effective in suppressing post-operative infectious complications. This slide shows the result of randomized control trials conducted to date using our symbiotics. This study demonstrates that the administration of perioperative symbiotics can improve the gut microbiota and intestinal environment and suppress the development of infectious complications. So why do symbiotics have efficacy against So why do symbiotics show efficacy against infection? First, I would like to talk about the association between the high invasiveness and the development of infectious complications. High invasive procedures such as surgery called dysbiosis and breakdown of the intestinal environment. So the concentration of organic acids, such as acetic acid and butyric acid, were decreased. We thought that this phenomena is associated with bacterial translocations. The next slide describes bacterial translocation. This biosis is associated with abdominal proliferation of infectious-causing bacteria, breakdown of intestinal barrier function, and decrease in host immune function. Furthermore, the infectious-causing bacteria migrate from blood vessels in the intestinal mucosa to mesenteric lymph nodes and other organs, and then spread in the entire body. This can lead to infectious complications. So, does the bacterial translocation actually occur? Using our gut microbiome analysis techniques based on RT-qPCR, known as scan, we show that bacterial translocation to the mesenteric node or blood occurred during surgery. That cannot be detected by a usual culture method. By detecting the bacterial translocation by if scan, we show that the detection of bacteria in mesenteric lymph node is strongly associated with the development of infectious complications. In addition, we demonstrated the occurrence of bacterial translocation in various gastrointestinal surgical cases, and this translocation is associated with complications. Therefore, bacterial translocation due to this biosis is significantly affect the prognosis in various pathologies. So, how do our symbiotics, our symbiotics effective in suppressing bacterial translocation? Here are the results. This slide shows the effect of symbiotics on the incidence of bacterial translocation. Significantly lower bacteria were detected in the mesenteric lymph node and blood samples in the symbiotics group than the control group. This result indicated that treatment with symbiotics inhibit bacterial translocation. So, how do symbiotics suppress bacterial translocation? There are two key points. First, symbiotics suppress the growth of pathogenic bacteria. Pathogenic bacteria, such as Acinetobacter baumannii, grew under condition reproduce the environment of patients without taking symbiotics. Whereas these bacteria cannot grow the condition that reproduce the environment the patient was taking symbiotics. And second, symbiotics enhancement the intestinal tight junction. In mouse infection models, symbiotic enhancement tight junction genes Claudin 1, occlusin, and Zone 1 than the control group, even compared with probiotics or prebiotics alone. Furthermore, acetic acid played a key role in enhancing the gene expression. This slide summarizes the preserved mechanism of the effectiveness of symbiotics. Bifidobacterium strain occurred utilize cracked oligosaccharide and producing acetic acid. Furthermore, obligate anaerobes in the intestine utilize lactic acid produced by lactobacillus paracardestrine silta and produce even more acetic acid and other acids. These acetic acid and other acids play a key role in inhibiting of abnormal proliferation of infection-causing bacteria and enhancing of tight junctions. Furthermore, symbiotics improve neutral state and recovery of peristaltic movement. In this way, symbiotics inhibited bacterial translocation and suppress the development of infectious complications. We have shown in other randomized controlled trials that symbiotics are useful for the side effect of cancer chemotherapy or reducing infection in clinical care settings. This suggests that our symbiotics may be useful for reducing the incidence of infection in various clinical settings. So this slide provides a summary of the presentation so far. Our symbiotics, lactobacillus strain silta, bifidobacterium strain Yakult, and galactorosaccharide have the potential to prevent infectious complications by improving the intestinal environment and suppressing bacterial translocation. In the future, we aim to expand the scope of application of symbiotics not only in surgery, but also in cases such as lifestyle disease. In addition, we hope to make improvements that will further enhance the inhibitory effect of symbiotics on infection. We will be happy to have opportunities to share these results to you. Finally, I would like to thank all our collaborators in this study. So that concludes my presentations. Thank you for your attention.
Thank you very much. Thank you very much, Sugimoto. Please stay for a couple of minutes. If there is any question, kindly respond them. Yes, Dr. Sasikaran, please. Any mic, kindly hand over to him.
Thanks for the good presentation. If we were to compare in these situations by giving symbiotics to some, by giving probiotics to others, by giving prebiotics to others, what would be the comparative effect? I know symbiotics would be the best, but what happens if we give only probiotics or only prebiotics?
Thank you for questions. But indeed, symbiotics, I think symbiotics is the best procedure. But one important point is in high invasiveness, and the administration of probiotic administration is through the intestine is very important. High invasive is called dysbiosis and the disrupted intestinal environment, and neutralization is very, very wrong. So the probiotics only alone administration is not well for lification. It's predicted. And prebiotics only, I think the commensal bacteria is very, very lower level. And especially characteristic nutrient bacteria is also very low. The prebiotic only administered. The eugenic bacteria is known that and the effect of prebiotics is very, very limited. I think very limited. So the symbiotics in our symbiotics is the probiotics and the probiotics can utilizing prebiotics combination. So such a long state, even such a long state, our symbiotics will work and improve this process, I think.
Thank you, Sugimoto. Thank you. You have any question? Please be brief.
Yes. I just want to know your recommendations regarding the timing and dosage of symbiotics for patients who are undergoing major surgery.
Sorry.
Dose of symbiotic and time of administration of symbiotic if patient is going for major surgery. What dose you recommend and at what time you are recommending?
Dose of symbiotic. Okay. Our oral narcotic RCTs and narcotic trial, the probiotics is dosed 100 billion cells each, and prebiotic characteristic is 6.6 grams.
I wanted to ask a question. Have you studied the role of probiotics Have you studied the role of probiotics or symbiotics in ventilator-associated pneumonia?
In ventilator-associated pneumonia, have you studied the role of symbiotic or probiotic or not? Have you studied or not?
Sugimoto-san, they're asking whether you studied the role of probiotics in ventilator-associated pneumonia.
Sorry.
Ventilator-associated pneumonia.
Sorry. In the last in the clinical setting, I will test the symbiotics in ventilator-associated pneumonia, and administered symbiotics is effective in infection, reducing infection in randomized control trial.
Okay, thank you very much. You can discuss when he is in the hall. You can sit with him and you can talk to him. That will be better, right? Okay, thank you very much, Sugimoto Sahab. Wonderful presentation, and let me compliment you for your best PPT. Your PPT was excellent. Your slides were very good. So thank you very much. Excellent presentation. Thank you.
That was quite a remarkable presentation indeed. I would now be requesting the chairpersons of this session, Professor A.K. Srivastava sir and Dr. Saurabh Nagpal, to kindly step forward and felicitate Mr. Takuya here. So please, if you could just kindly come back. I'm really, that's wonderful. Thank you so much. Can I just ask our honorable chairperson to kindly step forward and felicitate Mr. Takuya Sugimoto, sir, with a small token of appreciation from our end.
Your PPT was excellent. Very good. That was very good.
Thank you so much, sir. That was quite a wonderful presentation indeed. And everybody here sitting in the hall has been truly mesmerized by it. Please accept this token of gratitude, this token of appreciation from our end. Everybody, can we please have a round of applause, please? Thank you. Thank you so much. Once again, thank you, Mr. Takuya. It was really wonderful having you here with us. Thank you to our chairpersons as well. I would request them to kindly take on the lead on the next session now.
Coming to the next presentation, Dr. K. Kitahara. So he has done his marine microbiology at Kyoto University as an undergraduate student. He then moved to University of Tokyo to study molecular biology and biochemistry. He then worked at National Institute of Advanced Industrial Science and Technology and Osaka University as a researcher and became assistant professor at Hokkaido University. He is currently associate professor working at Collaborative Research Center of Okayama University for Infectious Disease in India, which is in ICMR NICED in Kolkata, India. I welcome him for his presentation.
20 minutes for you. Dr. Kitahara, 20 minutes for you.
Thank you for the introduction. And thank you very much for this opportunity of making a presentation about our research here today. I would especially like to thank Professor Ganguly, Dr. Hajira, and Chairman Professor Srivastava and Dr. Nagpal for this opportunity. The blue letters shown are the keywords for this presentation. Number 1, infectious diarrhea. Number 2, Vibrio cholerae and other enteric bacteria. Number 3, metagenome. And number 4, asymptomatic carriers. Before going on to the topic, please let me briefly introduce Okayama University's research collaboration with NICED in Kolkata. Okayama University is a university in Japan and has been collaborating with NICED for more than 15 years. 2 researchers, including me, are staying at NICED to conduct research on infectious diarrhea. And this is the name of our center. This is the collaboration scheme we are using in these 8 years. We are using this J-GREED program in which a Japanese university collaborates with a research institute specializing in infectious diseases. There are 10 such collaborations around Asia and Africa. This slide is about cholera and Kolkata. A strong point of conducting research in Kolkata is that it is easy to get cholera samples. Actually, Kolkata is at the center of the current pandemic of cholera. This is the aims of this study. The first one is to conduct metagenomic analysis of infectious diarrhea. We wanted to know microflora of both diarrhea patients and healthy people. To this end, we mainly used metagenomic analysis because it has a potential to detect all different bacterial species, including pathogens. The second one is to discover asymptomatic carriers of pathogenic Vibrio cholerae. Our hypothesis is that there must be asymptomatic carriers around a symptomatic patient. In the long term, we want to clarify the effects of such asymptomatic carriers. This is about the sample collection system from patients. We are using NICED surveillance system in which diarrhea stool samples are collected and brought to NICED. We first conducted conventional bacteriological tests as shown, and if pathogens are detected, we make glycerol stocks as well as a list of strains. And the original stool samples were always frozen at -80 degrees for metagenome DNA extraction. This is about how we collected healthy people's stool samples. This process was conducted by Dr. Kanungo at NICET. In the previous slide, we collected patient samples. When we get a patient sample, we asked the patient's family members or neighbors to provide their stool samples. The number of such healthy controls was 5 per patient. The samples were brought to NICET, and we extracted metagenomic DNA from them for sequencing as well as conducted conventional culture tests. This is a summary of collected samples in 1 year research period. Left columns are pathogens names detected by conventional culture tests. Next are the numbers of corresponding patient samples. And on the right are the numbers of healthy control samples. Because total number became too large for a single metagenomic project, we picked up a fraction with 10 patient samples and 65 healthy control samples. The 10 patient samples consisted of 1 enteropathogenic E. coli, 1 enterotoxigenic E. coli, 1 salmonella, 1 shigella, and 6 vibrio cholerae, or 1 samples. This is the strategy of our metagenomic analysis. We used 2 approaches. The first approach is to amplify 16S ribosomal RNA genes, and the amplicons were sequenced with a MySeq sequencer. The second one is shotgun approach, in which we just read the metagenomic DNA directly using a NovaSeq sequencer. First, this is the amplicon analysis data. Each sample's microflora was clarified like upper 2 figures. The left graph shows patient samples and the right graph shows healthy control samples. To summarize these results, we conducted 2 data analyses. The bottom left is alpha diversity analysis and alpha diversity analysis to know each sample's diversity. As you can see, healthy control stool samples had more diversity than diarrhea patient samples. Interestingly, however, some patients sometimes samples were as diverse as healthy controls like them. And they were found to be from patients with cholera. Another analysis is data diversity analysis to know similarity or relationships between samples. As you can see, healthy samples and diarrhea samples were well separated. But it was also true that some diarrhea samples were close to healthy samples, like this. Again, those samples are from cholera patients. What does that mean? Next, we used the shotgun metagenomic analysis data for further analysis. This heat map shows all 75 samples bacterial compositions. The upper tree shows similarity between samples. Upper right is the enlarged tree. We found that there are at least 4 clusters in the tree. 1, 2, 4. Cluster 1 was a cluster occupied by patient samples, and other clusters were from healthy people. By the way, the bottom right graph is an age plot of all samples. Cluster 2 was characterized by young age, and cluster 4 was diverse age, and the 3 was intermediary. In the tree, we can see that 4 cholera samples are in or near the cluster 1, but other 2 are somewhere else. Let's move on to further details. This is detailed analysis of diarrhea samples other than cholera. The left graph is for the EPEC sample, and it contained a large fraction of E. coli, as you can see. The right graph is for the Shigerosis patient, and it contained Shigella species. Because Shigella species are closely related to each other, species level identification may not be achieved. Anyway, pathogens were successfully detected. A characteristic was that pathogens were detected as a dominant population and diversity of microflora was low. On the other hand, this is a typical microflora of cholera patients. B. cholerae, Vibrio cholerae was only 2% of the total bacterial population, like this. Among 6 samples examined, only 2 showed Vibrio cholerae population over 5%. Others were less than 1%. This result means that Vibrio cholerae O1 does not necessarily occupy a major population in cholera patients' stools. This is an interesting discovery. And this is maybe why some cholera patients' microflora were close to healthy people's ones. And another interesting discovery is about asymptomatic carriers of Vibrio cholerae O1. We found 3 such asymptomatic carriers, 1, 2, 3, of Vibrio cholerae O1. And they were, we found 3 asymptomatic carriers, and they were family members of cholera patients. And these symptomatic cholera patients, this is the microflora of an asymptomatic carrier. Again, the population of the pathogen was low. The population of this sample was here, close to a symptomatic patient samples. You may think that the asymptomatic carriers were asymptomatic because Vibrio cholerae lost its toxicity. But it was found that cholera toxin A gene was detected from all Vibrio cholerae isolates. The right figure is the result of rabbit ileal loop test. It was found that all Vibrio cholerae isolates from symptomatic and asymptomatic carriers both possessed diarrheagenic ability. This is the summary of our study. First, 10 stool samples from patients with diarrhea and healthy, 65 samples from healthy people were analyzed. Number 2, respective pathogen was always detected by means of metagenomic analysis. Number 3, for patients with E. coli, Shigella, and Salmonella infection, each pathogen occupied the majority. Number 4, for patients with Vibrio cholerae O1 infections, the pathogen population was not necessarily large. And last one is 3 asymptomatic carriers of Vibrio cholerae O1 were found from healthy control people. One example showed that the proportion of Vibrio cholerae O1 was only 2% of all bacteria. And further studies would be to know the exact ratio of asymptomatic carriers of Vibrio cholerae O1. Currently, the number is 3 healthy carriers per 109 patients. And the second is to know why low amount of Vibrio cholerae can induce cholera. And the final one is to analyze why some patients become symptomatic while some are not. And that's the end of our presentation. Thank you very much.
Thank you very much. Thank you. Now, any queries, question? Yes, Professor Ganguly.
Guitar, a very nice presentation.
Thank you so much.
The For the first time, I see, we knew that very small number of salmonella can cause infection, but we never knew that very small number of Vibrio cholerae could cause infection. So this is a very important finding. I just want to know, I'm very worried about the human carriage of asymptomatic carriage of Vibrio cholerae, because these are small number. If you really do a population study, a large number of people who will be actually secreting the Vibrio cholerae O1, which are diarrhea, and can at appropriate time they can cause cholera. I thought the cholera is preserved in the environment, in the plankton, the blue-green plankton, and when the El Nino effect comes, they bloom and they cause cholera. But this is very significant, and I think you please enhance this study to find out. And the question which you are asking is very important, that what is the other microbial flora community in those who become carrier and those who do not become carrier. If the microbial community in those who didn't become carrier, we could understand, then we could perhaps populate those who are recovering from cholera infection with appropriate community of microbes to protect them from becoming carriers. I also find this metagenomic tool very good to really, I don't know what will be the cost of the Nova 6 shotgun analysis, but if I have to really do a epidemiologic study, I will use this because it is catching a lot of pathogen and their diversity, like the Shigella diversity, other diversity, nicely. So together you could get what are the pathogen and what are their diversity. But I don't know how much will it cost. So it must be costing a lot of money, but could it be made a simpler analysis, which is cost effective?
Thank you very much.
Very good. I learned a lot of things.
And our so metagenomic analysis strength is that we can how to say list up all suspected pathogens at once. So that quantitativity is very merit of using such metagenomic analysis. And thank you very much for your valuable comment.
Thank you. Thank you very much. Yes, please, quickly, one question, last question. Quick question.
From your metagenomic analysis, did you find any variation in the gene sequences of the cholera toxin gene? And if so, is it correlated with disease severity?
Yes, so we are now deeply analyzing using the shotgun metagenomic data. And from cholera patients, typical cholera toxin AB and other pathogenic factors are discovered. From patient samples, the situation is the same. So from such metagenomic analysis, we can also we can not only detect species pathogens name, but also specific pathogenic factor genes. So both we confirmed.
Okay. Thank you very much, Dr. Kitara. Yes, thank you very much. Wonderful presentation.
Thank you very much.
Please take a couple of minutes for some formalities, please.
That was quite a remarkable presentation indeed. And as I have our venerated chairpersons, Professor A.K. Srivastav sir and Dr. Saurabh Nagpal sir, presenting the memento or token of appreciation, of token of respect to Dr. Kitahara. Can we have a round of applause, please? Thank you so much. Thank you so much, sir, for coming in here today. It was a delight to have you here with us. And thank you to our chairpersons as well. Thank you.
So now next I would like to request Dr. Kriti Jain. She's a senior scientist working with Sir Gangaram Hospital and Research Center, New Delhi.
Dr. Kriti Jain, 20 minutes for you.
Next.
Sorry, it is only 10 minutes for you, Dr. Kriti. It is 10 minutes.
So a very good morning to everyone, respected teachers, chairpersons, seniors, juniors, and everyone present over here. I'm Kriti Jain, scientist, Department of Research at Gangaram Hospital. I'm here to present a talk on the role of gut microbiota in predicting response to checkpoint inhibitors in patients with advanced lung cancer. So first of all, coming to immune checkpoints, what are they? So PD-1 is a T-cell inhibitory molecule which is present on CD8 cytotoxic T cells, and PD-L1 is a PD-1 binding ligand which is present on tumor cells, APCs, and as macrophages. So when this PD-1, PD-L1 interactions takes place, it results in T-cell suppressions, which causes energy exhaustion as well as cell death. So the mechanism of immune checkpoint inhibitors is that the blocking of PD-1 and PD-L1 pathway would promote the function of exhaustic tumor-specific T cells and decrease tumor-induced immune suppression. So when this binding takes place, then the tumor progressions also decreases a little bit, and the antibodies, which are anti-PD-1 therapeutic monoclonal antibodies, like which we have studies as nivolumab and pembrolizumab, they act between these ligand and they enhance the therapeutic effect. So the rationale of the therapy is that of our study is that the checkpoint inhibitor therapies have got a huge revolution in the era of precision medicine as well as cancer therapies. But the only drawback, which are the clinicians which are facing right now, is only 30 to 35% patients response to this therapy. And this therapy has also many immune-related adverse side effects, and also they are very costly. Like one dose costs about 2 lakhs per dose. So the present challenge remains globally to develop set of biomarkers which can predict and monitor the responses of the therapy. And the present biomarkers which exist, these are the clinical biomarkers. They are undertaken on the tissue samples after the treatment. So these are PD-L1, tumor mutation burden, and microsatellite instability analysis. They are non-specific and they also have some limitations, like PD-L1 negative tumors, they are also responding to the therapy, and PD-L1 positive tumors also respond to therapy. So there is no consensus and there is no specific and sensitive biomarker which can predict the response to therapy and which can be used as a predictive biomarker to predict response. So the million-dollar question remains that the identification of specific biomarkers to predict response is the ultimate goal of clinicians as well as the researchers worldwide. So the aim of the study is to stratify cancer patients receiving PD-1 targeted therapy into 2 categories, responders and non-responders. So there are 3 categories as per the RESIST clinical criteria. This is called the RESIST criteria, which is progressive disease, responders, which are complete and partial, as well as stable disease. So to undertake the study, which we have taken the advanced lung cancer patients, both NSCLC as well as SCLC, and we have used these cohort in which we have taken PBMCs, the plasma, serum, FAPE block, fecal samples. From PBMC, we have done the extensive immune profiling using BD-Faxaria-3 and as well as BD-Canto. We have used plasma to do proteomics as well as metabolomic studies using mass spectrometry and ILBS. And from serum, we have done a cytokine study using FlexMec-3B system. From FFP block, we have undertaken genomic studies using ion Torin machine. And from fecal samples, we have done shotgun sequencing, 16S RNA sequencing using Illumina MiSeq. So in the total patients we have taken is 50, and we have taken samples before therapy at baseline, where when patient comes to the clinic and he or she is advised as checkpoint inhibitor therapy. So we have taken samples prior to the initiation of therapy, and after that we have taken samples at 2 time points, after 6 to 8 weeks and followed by another set of 6 to 8 weeks. So when the patient becomes responder or non-responder clinically, we have taken sample at the end point also as prescribed by the clinician. So we have done microbiome, immunomes, metabolomes, proteomes, as well as genomic study using this study. Due to time constraint, I will only be focusing on the microbiome and the metabolome associated studies that we have done. So coming to the plasma metabolomic study design, we have done this. We have taken plasma samples in discovery phase also, as well as validation phase also. And then at different time points, as per the RECIST criteria, we have categorized patient into responders and non-responders. And then we have developed a biomarker panel depending upon that. And also the statistical correlation we have done for the 5-year progression-free survival and 5-year overall survival of these patients using these parameters, using statistical significance of Cox regression using univariate and multivariate analysis. And we have also done the clinical correlation of the data with the various clinical associated parameters. So just this is a PCA and PLSDA plot, which is characterizing lung cancer patients with the healthy control. So here you can see a clear segregation between healthy and the patients. Then coming to the heat map and the hierarchical clustering analysis of the top 50 metabolites, which are capable to segregate lung cancer patients from healthy controls. So the top ones are the healthy controls at the baseline. And so this study was conducted majorly on the baseline to establish a set of predictive biomarkers which can predict response to the treatment. This is the volcano plot showing the characterization of responders as well as non-responders at the baseline. So these blue one, they are downregulated metabolites, and the red ones are the significantly upregulated metabolites. This is the PC and PLSD plot. So the reason why we have done metabolomic studies is that the gut microbiome, the major modulation of the gut microbiome is through the metabolites. So these metabolites, they are significantly present in the gut, and they causes anti-tumor responses to the local and the systemic presence in the gut, and they release secretory metabolites through which the immune response is generated. So this is the heat map also, which is showing the clear segregation of non-responders and responders of baseline at lung cancer. The red are the upregulated ones and the green are the downregulated ones. So now coming to we have also done the metabolomic pathways, which is the top 3 pathways which we have identified is the overview. This is the overview of enriched metabolite set, which is the top 25. So giving you the glimpse, we have identified as for the basis of the significant pathways which are associated with the metabolites, which is sphingolipid metabolism, arachidonic acid metabolism, as well as the glutamate acid metabolism. So sphingosine, this induces tumor cell proliferation, resistance to chemotherapy, radiotherapy, and immunotherapy. This is known, and it is published in Nature and several other papers also. And as well as the glutamate metabolism, this reconditions tumor microenvironment, which can enhance anti-cancer immune responses through both direct and indirect mechanisms. So there are various studies by which we can correlate our results, which we have got in this metabolomic studies. So this is also a cake pathway analysis of the metabolite sets, which are top 25. And now, based on this analysis using the discovery set as well as the validation set, we have identified this panel of 5 biomarkers, which is hydroxybutanol, gentasic acid, urea, sphingosine, as well as the glutathione. So these 5 biomarker panel, they successfully predicted response to the therapy at baseline, and they are responsible for generating good response. So glutathione is also most abundant antioxidant found in living organisms and prevents sufficient level of cysteine and detoxifies xenobiotics and also conferring therapeutic resistance. So when these glutathione, except from urea, everything which you can see here is enhanced in responders, and the only urea is the biomarker which was clearly characterizing the non-response. So urea cycle dysregulation, this generates clinically relevant genomic and biochemical signatures. So urea cycle dysregulation increases the pyrimidine and purine imbalance, which resulted in mutation bias. So as per the hypothesis of the checkpoint inhibitor therapy, more the mutation bias, more the antigens would get antigen binding sites where it can bind. So more the monoclonal antibodies will get site to bind. So more the mutations, more the hotspots, and more the therapy should work. So this urea cycle dysregulation is primarily creating this mutation bias, which is increasing the number of mutations, and thereby the therapeutic response and the efficacy of the immune checkpoint inhibitors is increasing. And same, the role of sphingolipid metabolism is a signified role in the drug cancer resistance. So it was also enhanced in non-responders, which is giving this result. So now coming to 16S microbiome analysis. So first of all, we did alpha diversity analysis. This we have done from the fecal stool samples, and then we have isolated DNA. And then we have categorized two subsets of population based on the RESIST clinical criteria, responders and non-responders. And at this study was also done at baseline. So this is the box plot showing alpha diversity index, observed O2U, evenness, Shannon index, and Chow index between responders as well as non-responders. So we did not get any significant difference between the two groups. However, the responders and non-responders showed a clear segregation. So now, based on the Wilcoxon test, we categorized what are the differentiating bacterial population which may be present persistently in responders and which may be present persistently in non-responders, which can give us an idea that whether the patient is going to respond or not respond. So this gut microbiome, it is playing a very complex interplay between the clinical parameters as well as the metabolomes, as also after the therapy, the patient is getting enhanced CD8 cytotoxic T cells. So this complex interplay is very important to be studied, and no single bacterial population, what I have studied till now as per the recent literature, no single population can give us the idea that whether the patient is responding or not responding. So we need to study these in larger cohort. So as per our data, what we have got is, we have got that Bifidobacterium and Doria species, they were significantly enhanced in responders and they were absent totally in non-responders. So Bifidobacterium, as we all know, is a healthy bacteria which is present in the gut, and it is also causing augmenting the response of the antibodies and the monoclonal therapeutic drugs that we have given, which is nivolumab and pembrolizumab and Klebsiella, Enterobacter, and these families, these genus were persistent in non-responders at baseline. So this is a very, very interesting and significant data which we have got, that even Klebsiella is known to be causing drug resistance in wide number of solid tumors as well as melanomas and lung cancer and breast cancer. The studies have been conducted which have shown that the Klebsiella species is persistent in the resistance population of chemotherapy as well as immunotherapy. Also, Bifidobacterium is a known bacterial population which we have got. Also, we have got Porphyromonas and genus family 13, which is not named yet, but these were persistently higher at baseline in all the responders population who responded well clinically, and they were clear-cut responders. They were not partial or stable responders. They were complete responders who showed this bacterial population high at the baseline in the population. So this is the pie crust GitHub analysis using Wilcoxon test, p-value less than 0.05. We have taken the pathways which are associated with these bacterial populations. So we have done the sample-wise abundance distribution also, as well as the mean abundance distribution between the groups. So this is the pathway analysis, and the gallate degradation pathway, hydroxyphenyl acetate degradation pathway, catechol orthocleavage pathway, they were associated with the responders, and they were also present in the mean abundance distribution between the two groups. So this is the table which shows the differentially abundant core functional pathway by using PyCrust2 analysis GitHub, Wilcoxon test, and p-value was also significantly high. So the pathway which is associated with amino acid metabolism, which was shown to be higher in responders, and that was downregulated in non-responders. Also, the xenobiotic degradation pathway, it was significantly high and upregulated in all the associated microbiomes in all the populations in the non-responders. So what we have done next, we have tried to done a correlation analysis in the metabolomes and the differentially abundant genus in responders and non-responders separately. So the correlation coefficient which we have taken here is more than 0.5, which is indicating a strong positive correlation, and less than 0.5, which is indicating a strong negative association. So from the metabolites data, we have found out that we have tried to correlate the results of the microbiome. So these here in this table, you can clearly see that the benzaldehyde is showing a positive correlation, strong positive correlation between the family 13, and the linoleic acid is showing a strong positive correlation between the Bifidobacterium. So what we mean to here say this, that if we critically look at these population in the starting, and we can modulate the gut microbiome some way, as it was being talked about yesterday in several presentations, by giving some fecal microbiota transplantations or genetically engineered microbes, and several other techniques by giving probiotics, of course, and prebiotics at the initiation of the treatment, if we can overcome these bacterial species population and if we can regulate these metabolites in some way to enhance the therapeutic response, these 30 to 35% of patients may change to more than 50%. So that will be also a challenging thing. So here we should focus on, and a larger study should be taken out in this area. And even if you see at the responders where there's a strong negative correlation between NAD and Doria and NAD and porphyromonas, these patients were also responding well to treatment. So next, what we try to do is we try to establish a correlation network between significant microbes and metabolites in responders versus non-responders to checkpoint inhibitor therapy. So the threshold value here taken is again 0.5 and minus 0.5. The edges indicate positive correlation, whereas the blue edges indicate the negative correlation association. So the width of the edges between the nodes and indicate the strength of the association. So if we critically look at these, then we can definitely get some set of biomarker panel and biomarkers which can predict response. So this is the overall heat map showing an overall correlation matrix using Spearman's correlation coefficient between differentially abundant genus and metabolites in responders and non-responders. So there's another description and another representation of what I've just shown to you in the slide. So these are the different categories which causes and which I just talked about, that these can be used as an interventional measures to improve the efficiency of cancer immunotherapy. These papers have been published very recently in Nature as well as Lancet, wherein the gut microbiome modulation plus checkpoint blockage, if you give these drugs along with the gut microbiome modulation, if you do 16S RNA sequencing along with the next generation sequencing, yes, I know that they are very costly, but we can try to make some biomarker panel, and if they can, if the patient can benefit from these, it will be a very good, and it will be a good era in the new precision medicine of oncology. So studies have shown that yes, if you give checkpoint blockage along with the gut microbiome modulation as per the treatment basis on the patient to patient thing, then cancer outcomes have improved, therapeutic outcomes have improved, along with probiotics, prebiotics, and these papers have shown bifidobacterium, lactobacillus, as well as using the prebiotics such as resistance charge and inosine, which just was shown in the presentation, that the therapy outcomes have improved. So as per the timeline, which I will just show you, that there are several studies which have been ongoing, which shows that how gut microbiota could stimulate anti-tumor immune responses and antibiotics attenuated effects of cancer therapy. So since 2007, mouse studies have been done since 5 years, 2007 to 2013, as well as in 2015, preclinical studies are also being done in mouse model, which has shown that the therapeutic response have enhanced in the mouse model, which has bifidobacterium species. Also in 2018, some studies have indicated that the role of gut microbiome and fecal microbial transplantation in responders versus non-responders. And very recently, 2021, 2 clinical trials, Keynote 46 and Keynote 061, have clearly shown that these FMT and probiotics, yes, they have overcome the resistance to immunotherapy, and they have enhanced the therapeutic effect after the treatment. So these are the recent papers showing that how the gut microbiota modulation enhance effect of checkpoint inhibitor therapy, and metagenomic analysis, which is revealing distinct gut microbiota signature for predicting adjuvant therapy and responses in patients. And this is a very good article which has been recently published, which shows in Nature that cross-cohort gut microbiome association with immune checkpoint inhibitor response in advanced melanomas. So in this paper, they have also identified bifidobacterium and Doria species, same as we have identified in our responders group in melanoma, and we have identified these species in the lung cancer group also. So this will be a very interesting study if we can conduct in the larger cohort. So I will just give you a glimpse of targeted proteomics results that we have got using the Olink platform. This work was done at Karolinska Institute, Sweden. So in this Friedman's test showed significantly changed proteins between the 3 time points, baseline, and subsequently at 2 other time points. So IL-13, granzyme A, VEGFR, granzyme H, CD27 was significantly higher in non-responder, whereas granzyme B, CRTM, and CCL23 was significantly higher in responders. So granzyme B is a cytotoxic secretory T cell molecule, which we have got in our flow cytometry results also. This granzyme B we have got in the cytokine population also. So this exhibits cytotoxic effects on the cancer tumor cells, and by which ultimately the response to therapy is increasing. So these are the temporal changes that we have observed from baseline to time point 3 in
Please
various
wrap up,
groups.
Dr. Kirti.
Yes, sir. Yes, sir. So these are the temporal things. I will just shortly give you an idea that using this biomarker panels of metabolites and microbiome, we have done this ROC curve analysis using and predicting sensitivity and specificity. This is the Kaplan-Meier analysis of categorizing 5-year progression survival in lung cancer patients using these parameters. And also we have done the Kaplan-Meier analysis of 5-year overall survival of these lung cancer patients of using this predictive biomarker panel. So just to summarize my work, so however, I've not shown the entire data that what biomarkers we have got in different subsets of different biomarker category, but I will just summarize to you that immune cells, we have got that CD4 helper T cells, CD8 helper T cells were significantly enhanced using after therapy in responder subsets. Also, the proliferation of cytotoxic T cells using Ki67 were enhanced. Also, we have got the NKT cells population, which was significantly enhanced in the responder subset. As well, when we come to cytokine expressions, then responders IL-2 and 23 was enhanced in baseline, and IL-18 were enhanced also at the responders. So focusing on the gut microbiome signatures, as I already said, Enterobacter and Klebsiella were persistent in the non-responders, and Bifidobacterium were there in the responder subsets. So just to shorten and just to summarize and give you everything in together, so this multi-omic approach along with comprehensive biomarker phenotyping may serve as a potential biomarker panel to monitor or predict response to checkpoint inhibitor therapy. And this may broadly categorize patients receiving checkpoint inhibitor therapy into 2 categories, responders and non-responders. So this mechanistic exploration would provide novel insights for developing rational to microbiota-based therapeutic strategies by manipulating gut microbiota, such as FMT, probiotics, engineered microbiomes, and specific microbial metabolites to augment the efficacy of ICIs and advance the age utilization of microbiota in precision medicine. So I would like to acknowledge many people and also Professor N.K. Ganguly sir for giving me this opportunity. And he was the overall mentor in my entire work. And Professor Amit Avasthi from THSTI, Dr. Shyam Agarwal, Dr. Rashmi Rana, Dr. Jaswinder Singh Maras from ILBS, Dr. Bhabtosh Das from THSTI, where all the microbiome work was carried out, Dr. Surajit Ganguly from Jamia Hamdard, Dr. Deepak, and Ms. Parul Chuk. Also, I would like to acknowledge the faculty of Department of Research, Dr. Jill Neogi, Ms. Deepa Mehra, Dr. Daisy, Medical Oncology Department, SGRH, and these are the funding source. And here you can see a complete responder picture. So this patient had come with the buccal cavity mucosa, and just after 3 doses, the patient was completely treated with the nivolumab monoclonal antibody. And this result correlated with our disease outcome and with our biomarker panel also. So thank you very much for listening.
Thank you very much, Dr. Kirti. Thank you. Friends, as we are running short of time, so if any questions that can be asked to her during tea time. Thank you very much. Thank you.
Thank you, sir.
Stay for a minute on the stage.
Truly a wonderful presentation indeed. Can we have a round of applause for the lady here, please? And our chairpersons are stepping forward to felicitate our speaker here with. Ma'am, please accept this token of appreciation from our end. It is a delight to have you here with us. Everybody, can we please have a round of applause? Thank you so much. Thank you, sir. Thank you, ma'am.
World has been taken over by tech world and marketing else. This is the place where funding is required. This is the place which requires more efforts. These days, people pour in Swiggy's, Zomato's. This is the place where funding is required. Delivering a food in 5-10 minutes is pretty secondary, I feel. This is the place where exact effort is required from the world. Our next speaker is Dr. Caroline Childs. She is a registered nutritionist, is associate professor of nutritional sciences at University of Southampton, working within the Faculty of Medicine. Dr. Child is nutrition and metabolism subject lead for undergraduate medicine and program leads of BM6 widening participation undergraduate medical degree. Dr. Childs is a Southeast regional representative for Association of Nutrition. Her research expertise include the effects of dietary fatty acids, probiotics, prebiotics, antioxidants upon the immune system. Dr. Child's experiment approaches have encompassed assessments of tissue composition, immune function, inflammatory status, and immunosenescence. I would like to request her to be here to proceed with her presentation. It's for 20 minutes. You can start.
Very much. Can I just check? Can you hear me all right and see my slides? Okay.
Okay. Yes, your slide is.
Yeah. Great. Okay. Well, thank you so much to the organisers for inviting me to speak with you here today. My offices are based in Southampton in the UK, and this is a picture of my offices on a nice sunny day. And I'm delighted to work with a fantastic team of nutritionists and people who focus on the immune system as I do in my work. So in my talk today, I'm going to talk to you a little bit about the role of probiotics on the immune system, particularly as we age. So the areas that I plan to cover are, first of all, how your immune system ages and how people like me can measure that in the laboratory. And then I'm going to talk a little bit about the evidence I think there is so far for probiotics in aging adults. And I'm going to talk a little bit about the quality of those studies and also the latest reviews that are available for you to read if you would like to learn more about that topic. Okay, so aging of the immune system is something called immunosenescence, and this is where our immune system, both the acquired arm of the immune system, that's the type that helps you in vaccination responses, for example, and our innate immune system, our first line of defense, both become deteriorated and not functioning in the way that they usually would. Now, in my talk today, I'm going to focus on the effects on T cell function. That's an area which has been quite well categorized in the research, but I just want to highlight that it would also be possible to do a talk on B cell function or many other types of immune cells as well. I'm going to talk a little bit about natural killer cells as well today. But this is important because we know that having a poor T cell response is linked to increased morbidity and mortality, and that we, as people age, we see that they start to become more pro-inflammatory and perhaps have a chronic low-level inflammation running in the background all the time. Now, your immune system is complex. It involves a range of organs, specialized tissues, and specialized cell types. So one organ that we understand changes with age quite clearly is our thymus. So this is where your T cells mature, a small organ sitting just in front of your heart. And this is at its peak in childhood. Now, each time I do this talk, I find myself moving further to the right on this scale. I'm around here now. And what happens with age is that the functional tissue within your thymus decreases. So for me now, my thymus is probably mainly made of fatty infiltrates with much less remaining functional tissue than I would have had in my childhood, for example. So this is a sequence of aging that we see occurring. And I suppose one of our questions is there anything we can do about this? And when would be the time to do something about this? Now, the reason that matters is that when you have a young thymus, what happens is you're exporting a wide range of diverse T cells from your thymus. And you have relatively few memory T cells because you haven't been exposed to many infections or challenges at that stage in your life. As we age, we start to have a reduction in our export of these naive T cells, randomly associated to be able to respond to a wide range of pathogens. And eventually, as we become much older, we're starting to get this increasing pool of memory T cells. So we may have less diversity in our new immune cells which are being produced. Those memory T cells may become less effective, even if they do recognize something. And so this can be a major challenge. And this was something that all of us experienced and observed during the COVID pandemic, where older adults were much more likely to have severe symptoms or to be at risk of death as a result of COVID compared to younger adults and children. Now, while aging is inevitable for all of us, I just want to highlight that there is some variability in how that aging happens for us. So as I look at this plot here, this is one marker of immune cell aging. And as we look with age, we can see that this declines steadily with age, with a particular drop-off after about the age of 50. But one of the things I want to highlight to you is that there are some 70-year-olds with an immune system as good as the average 20-year-old, and that there are some 20-year-olds with an immune system as poor as the average 70-year-old. So one thing I would really encourage is for us to start moving beyond thinking about just chronological age. There are ways that we can assess someone's risk by looking at their relative immune age in the laboratory. As I mentioned, older people are much more likely to get infections and have more serious complications associated with this aging. And this is clinically significant. So, for example, in terms of vaccination responses, there were some estimates made in the UK that a significant number of deaths associated with influenza and pneumonia, those occur in those over the age of 65. But even with an established vaccination program, a significant number of older adults, their immune system is so impaired that even with vaccination, that doesn't provide them protection. Now, to be clear, I'm totally in support of vaccination programs, but we just need to be aware that not everyone has a good response to those vaccinations because of the aging of their immune system. Now, one of the key drivers of your immune system is your gut microbiome, and I'm sure I don't need to convince the audience here of the importance of the gut microbiome. But one of the things I just want to highlight to you is how that links with age. So we know that our gut microbiome changes based on the diet that we eat, and our diet changes significantly over our life course. We go from an infant with an exclusively milk diet to having a more diverse diet, but perhaps in the later stages of life, our diet diversity may reduce because, for example, our meals may be being provided for us, we may always be eating in the same setting, and perhaps having a pattern of meals which is repetitive in later life. And so various studies have demonstrated this. This was a study looking at older adults in various residences, so those who are living in the community or in rehabilitation facilities having one particular type of microbiome, whereas those in long stay residential care having quite a reduction in diversity and quite a different profile of gut microbiome. But one of the challenges is that not all older adults are the same. You know, if we talk about the over 65s, this is not a homogenous group, you know, so we can also further break things down by decades, for example, and we can see those changes in the microbiome with increasing age. And also we can look at where someone has been residing and how long for. Those are all significant predictors of the gut microbiome. So that's one of the challenges in doing aging research. If I say I'm researching older adults, am I talking about my latest study looked at adults aged 50 to 65, or am I talking about a study which looked at people over the age of 85? Those would be quite different pieces of work. One of the other things, of course, important for us to remember is that the species and strain of probiotic that we provide has significantly different effects on immune function. And I think all of us in the audience are probably familiar with the various species and strains of probiotics which are available. These are just a few examples. But one of the things I want to highlight to you is that as well as having those differences between species and strain, it also depends on the patient population that you are providing this to. And this was a study which was done in healthy, young and older adults. And you can see that the same strain of a probiotic can have quite markedly different effects depending on whether it's being provided to a young or an older person. And so, for example, for some strains, there may be no effect, no significant effect in some groups, but a very significant effect in others. So I think another thing for us to take care about is considering the population that we're studying. I'm interested in aging. Perhaps here on this graph, old is not enough information. You know, what type of older adult are we trying to study here? And so that's just something I also wanted to highlight to you. Now, in terms of what I aim to do in my own research, I use flow cytometry quite a lot. It's one of my favorite methods. And so in doing that, what I can do is so for example, my current human study, we have participants coming in, taking supplements, we collect blood samples, and then I can look at specific immune cells within that population. So I can identify lymphocytes, monocytes, and neutrophils. I can identify T helper cells, for example. And then what I'm particularly interested in is drilling down a little bit deeper and looking to see the relative age of those T helper cells. And so I can differentiate these into populations of naive T cells, those which are effector memory T cells, those which are central memory T cells, and those which we might call terminally differentiated T cells. And in younger adults, you will see almost none of these. And then as people age, these start to accumulate. And so this accumulation of these and the decline in the number of naive T cells is a classic feature of aging that we see. And so with techniques like flow cytometry, I can look in detail at the markers that are produced by these cells, the kind of cytokines they're producing. I could isolate them and do some interesting mRNA sequencing. So there's a whole range of things we can do looking at the relative age of someone's immune cells. So I'm just going to highlight a few of those markers that I'm particularly interested in. One of them is CD28. So my image of myself is just blocking my picture here. I'm just going to move that a little bit. So CD28 is one of these cell surface receptors, which is important in these T to B cell interactions, having this interaction here. So it's what's known as a secondary signal, allowing a strong co-stimulation of your immune system. And this is something that we see expressed on T cells, but this is something which is lost with aging. Okay, so amongst cytotoxic T cells, those CD8 T cells, amongst older adults, they express this CD28 on less than 10% of their cells, whereas a younger person will have that on up to 75% or more of their T cells. So this is a marker I can use to assess someone's relative aging of their immune cells as well. Another marker which we can look at is CD57. This is something which is found expressed on T cells and natural killer cells. And again, this is another proxy marker or biomarker that we can use to look at aging. The accumulation or the expression of CD57 is something which is associated with an exhausted immune cells. And so we can characterize cells which are immunosenescent or aged by their lack of CD28 and their higher expression of CD57. And various studies have looked at this, and I just want to highlight how much of a significant predictor this can be of health outcomes. So I'm particularly interested in nutrition and the immune system, but this is some example data where they looked at the presence of these CD58, CD57 high cells, and this was on cytotoxic T cells, and they found that amongst people who had their first acute MI incident or heart attack, their survival over the days which followed was significantly predicted by the presence of these aged immune cells. Okay, so this is something which our immune system has functions, of course, across our whole body. And so there are a whole range of health conditions which I think can be examined by us understanding this aging of our immune system. Another marker which I'd like to bring to your attention is something called Tregs, or T cell receptor excision circles. So when your immune cells are being generated, there's lots of gene rearrangement taking place to make those receptors for various pathogens. And during that process, small circular regions of DNA are produced. And so within a naive T cell, there'll be a number of these circular regions of DNA associated with the assembly of the T cell receptor. Now, because that is not, when that cell replicates or divides or proliferates, that circular DNA does not replicate or divide. And so with each division of cells, those number of T cell receptor excision circles will halve. So in a brand new cell, you'll have a high number of these T cell receptor excision circles. And in a cell which is perhaps older, many generations of division have occurred, you'll have much fewer of those. So this is another way that we can assess the relative age of an immune cell. And this was a study I was involved in at the University of Reading. What we did was we had volunteers who were either older adults, in this case we were looking at those 60 to 85 years of age, or younger adults. In this study, we classified younger as being 18 to 35, but I remember feeling quite uncomfortable when I realized I was no longer eligible for this study. And what we did was we provided these participants with, it was a placebo-controlled trial or a symbiotic study, which in this case was a Bifidobacterium lactis and a gluco-oligosaccharide. So people took this symbiotic for 4 weeks and then had the flu vaccination, and then we followed them up for another 4 weeks to look at the effect on their immune cells. But one of the things I want to highlight to you is this is at their baseline data, the number of those T cell receptor excision circles that they had. And I just want to highlight to you, you can see that younger adults have significantly more of these compared to older adults. But again, to highlight that overlap, there are some older adults with as many as the average young person and vice versa. So there's a great deal of variety in studying our immune responses. And that can make it quite challenging for me in the laboratory because I can see people are very different from each other. Even when I've tried to, for example, stratify my recruitment by age, by sex, by BMI, for example, we need to take into account this immune age to understand the effect that our supplements or products may have upon someone's immune system. Here's another example of some of the data which came from that study, just showing that this immune aging matters. So here, this was looking at those cells that had lost CD28, had gained CD57 on those T helper cells. And here in this study, we were looking to see amongst our participants, which of them had achieved seroconversion to the vaccine. So which of them we would consider to be protected post-vaccination amongst our older adults. And what you can see is that those who had the most loss of CD28 and the most gain of CD57 were significantly more likely not to convert in response to vaccination compared to those who successfully achieved seroconversion. So these are important factors for us to consider in targeting healthcare systems as well. Now, for the purposes of time, I've focused mainly on those T cell responses. But again, I just want to highlight to you that there are a whole range of markers we could look at to assess immune function. So as I said at the beginning, we could look at things like B cells. We could look at things like exposure to cytomegalovirus. And I suspect in research to come, we'll be looking at markers of exposure to COVID, for example, as well, in terms of aging processes. But I just want to highlight to you that all of these factors come together in what some authors have described as an immunological risk profile. And so can we identify you or I's immunological risk? And are there any steps we can do once we know what our risk profile is? So just to come on to some of the evidence around probiotics, there are systematic reviews available for the effect of probiotics on immune outcomes such as upper respiratory tract infections. And these studies have identified that probiotics are better than placebo at reducing the number of episodes of upper respiratory tract infections, at reducing use of antibiotics amongst children, reducing school absences. So in that regard, the evidence seems quite strong for probiotic interventions. But one of the challenges we have in this type of nutrition research is how our evidence is rated in terms of its quality. And in this paper, for example, they noted that there were few risks of probiotics and relatively mild GI symptoms are the most commonly reported side effect, but that there is a high level of variety or heterogeneity of that data, and the evidence level was rated as being low or very low quality in this systematic review. So this is a challenge for all of us working in nutrition research to ensure that our research is rated as being of good quality, but there are some challenges associated with that within nutrition research. But assuming that these effects were, sustained, what kind of economic impact might that make? And this is some work which modeled that health economic impact of probiotics. And what they modeled was if everyone in a model population of 50,000 people was taking probiotics over the typical peak period for cold and influenza-like illness, October to April, what effect would that have on health outcomes? And they identified that it would reduce a significant number of episodes of respiratory tract infection, would reduce a number of antibiotic prescriptions that were made, but perhaps most important in persuading policymakers is the effect that it has on productivity and ability to attend work. They estimated that this would reduce 1,400 sick days. And when they modeled that in terms of impact on the NHS, in terms of appointments and prescriptions, there were savings, of course, to the health service, but much more significant savings for society as a whole for avoiding those number of sick days, which many of us may have over a typical winter period. Now, I did a review of the available evidence that there was for the effects of probiotics on the immune system across the life course. And I feel that there's quite good evidence that probiotics can increase vaccine responses, have particular effects on natural killer cell activity, but also effects on other measures like phagocytosis and oxidative burst, those innate immune outcomes, and measures of infections or symptoms of infections. But one thing I would note is that of these studies that I looked at, only 2 of them used markers of immunosenescence. One of them was work that I was involved in, and so only one other group had measured this. And also there are challenges with the quality of that evidence. 10 of those studies were single-blind, open-labeled, unrandomized or uncontrolled trials. And this will always be the criticism that we might get in the nutrition field if we don't correctly design our studies. But I do acknowledge that there are significant challenges for us in nutrition research in doing that. How do we blind a commercially available product when we're intervening with it? If we're designing an intervention and it's something where similar products are available on the market, how do we control for background intakes of that product? We're not designing a novel drug that no one else has any access to. If we start recruiting people to a probiotic study and they start to feel that may be beneficial, how can we can't prevent them from also buying those products in their own life as well. So there are unique challenges to doing nutrition research, but it's all of our responsibility to try and do the best studies that we can. So I mentioned that one of the studies that had looked at these immunosenescence markers was my own, and I showed you some of that data earlier. But just in terms of other outcomes that people have found from probiotic supplementations, this is the work of Maura Garcia in 2013. They found that by providing this probiotic, and they were looking at older adults 65 to 90, average age of 70, that this increased the number of natural killer cells and those naive T cells, for example, and they saw that the cells looked relatively younger by their T cell receptor excision cycle expression. Now, I just want to start wrapping up by highlighting to you some of the other evidence which is available for you to read. So here's a recent systematic review that was looking at probiotics and those effects on the immune system with a particular function on natural killer cell activity. And again, they identified that probiotics significantly increase natural killer cell activity. But again, we face the same challenge. There was a small final number of studies, very large heterogeneity. And so these authors concluded that this evidence was not convincing. So this is something for us to consider. Another review I'd like to bring your attention to was looking at probiotics and prebiotics on aging as a whole. So they weren't just looking at immune parameters, but also things like blood glucose control, cognitive function, gut microbiome. And again, they found that they rated a significant number of these studies of having a significant risk of bias, either due to changing from the intended intervention, for example. So another challenge for us to consider in our research design. So I'll just conclude by saying I think it's very well understood and a key area of research that there are these interactions between our gut microbiome, our immune system, and how we age. And I would suggest the evidence for probiotics on natural killer cell function is very well established and has been seen in multiple human intervention studies. I hope I've shared with you some details of some of the techniques that are available for us to assess immunosenescence, and that there is a small number of studies, but some which have provided evidence for a role of probiotics in that process, but also for us to be conscious that there are significant challenges for us in obtaining high quality research evidence in the field of nutrition research and immune function, and that current systematic reviews are consistently pointing to our variability between studies, the low quality of evidence, and the high risk of bias. So I'd just like to finish up by acknowledging my funders that I've had over recent years, and also the fantastic work of my colleagues. So it's a great privilege to work with this team in Southampton. And thank you very much for your attention today and for inviting me to speak with you all. Thank you.
Thank you very much, Dr. Carolina Child. Well, friends, these are all 6 good presentation because we are already very late, running shortage of time. So we will not take the time to repeat the things what they have spoken, but it was a wonderful session, wonderful presentation. We enjoyed, and there was a lot of addition in our existing knowledge. I will request Dr. Saurav to say just a few words.
It was great to have all the presentations. Thank you very much to all the presenters for enlightening us and presenting us with the in and out of the existing chemical modules. Thank you very much.
Thank you, Dr. Carolina Childs. Thank you. It was a wonderful presentation. We enjoyed. Now you can you can have sleep, right? Thank you very much. Thank you. Thank you.
This brings us to the end of our first session here with, and can we have a round of applause for our dignitaries here on stage, our chairpersons who diligently curated the entire session. I would now be requesting the humble presence of Dr. B. Sasikaran to please join us here on stage and felicitate our first chairperson here with. Can we welcome Dr. Sasikaran with a round of applause, please? Please come, sir. And I would request Professor A.K. Srivastava to kindly step forward and accept this token of appreciation, this token of respect. Everybody, can we please have a round of applause for sir here? Thank you. It was a delight to have you here with us, sir. Thank you so much. I would also like to invite Professor N.K. Ganguly, sir, to please come on the stage and present token of appreciation to our chairperson, Dr. Saurabh Nagpal. Welcome, sir, on the stage. Professor Ganguly would now be presenting the token of appreciation to Dr. Nagpal. Thank you, Dr. Nagpal, for your eminent time here today. It was wonderful to have both of you here for conducting the session in such a fantastic manner. Thank you. I would request the photographer to please step forward for a group photo op here with. I didn't take it. Thank you to the dignitaries. We will now be taking a small tea break, and I would like to remind you all to please be back here effectively in 10 minutes. As you are running a bit short on time, we would want you to be here in 10 minutes for our next session on young investigator presentations. Thank you so much. Good night, patients.
हाँ कर देंगे।
Hello, sir.
You can hear now.
Yes,
It was
sir, I can
muted.
hear you.
It was
Right
muted.
now, the tea break is going on there.
Oh, okay, okay. That's why it's muted. I saw somebody on the podium. It was muted. That's why.
It will start now, sir.
Okay, no problem.
Okay, sir.
Right.
Welcome once again to all our honorable guests. Thank you so much for coming in here. The tea break is officially over. We now begin with our next session, which is Young Investigator Presentations. The chairpersons for these sessions are as follows: Professor J.B. Prajapati, sir, and of course, Professor J.B. Tamang. Let me enlighten you a bit about our dignitaries here on stage. Professor J.B. Prajapati is a dairy technologist from Anand with PhD in dairy microbiology from NDRI. He retired as the Dean of the Faculty of Dairy Science at AAU Anand. He's the Chairman of Indian Dairy Association, Gujarat State, and coordinator of Sasnet Fermented Foods. He's a renowned academician and scientist in the areas of probiotics and fermented foods. He has more than 500 publications, including 2 books. He had Fellowship Award from the Erasmus Mundus EU as well as Indian National Science Academy. He's a fellow of Indian Dairy Association as well as National Academy of Dairy Sciences. At present, he's the chairperson of Fergus Curran Center of Excellence at Institute of Rural Management, Anand. Can we have a round of applause for Sir here, please? Thank you so much. Next, I would like to enlighten you a bit about Professor Dr. Jyoti Prakash Tamang, who is a food microbiologist of fermented foods. He's a fellow of Indian National Science Academy, National Academy of Sciences India, National Academy of Agricultural Sciences, Indian Academy of Microbiology Sciences, and recipient of Bioscience Award of Department of Biotechnology. He has published more than 190 research publications, including 8 books, 28 book chapters with more than 7,800 citations and 50 H-index. Professor Tamang is the senior professor and head in Department of Microbiology, Sikkim Central University, and is also the chairman of scientific panel of Alcoholic Beverages of Food Safety and Standard Authority of India. Can we have a round of applause for Sir here as well? I would request our honorable chairpersons to kindly take the lead and conduct the session forward. Thank you.
Thank you. At the onset, I thank the organizers for giving us opportunity to chair the session. As all of you know, we had a big poster sessions, and after the first round, we selected 20 topmost. Those are displayed outside, and from that we had the 3 best posters, and these 3 presenters will be presenting it today. I have pleasure in inviting the first winner of this poster session. Is jointly written by Krupali Ramanujan and Dr. Subrato Hati. So Subrato Hati is presenting today the first winner poster position. Subrato Hati is assistant professor at the Department of Dairy Microbiology at Asmisi College of Dairy Science at Kamden University, Anand. And he is one of the very renowned scientists in the area of probiotics and bioactives. And he has a number of awards to his credit. And we'll be happy to hear you, Dr. Subrato. I will request you to finish within inside 10 minutes, less than 10 minutes, I will say. Thank you.
शेयर करना है तो करो ऑनलाइन में भी होगा ना यहाँ से डायरेक्ट हो जाएगा चेंज करना है तो
इधर आ गया. A very good afternoon to the chairpersons of this session, Dr. J.B. Prajapati and Dr. J.P. Tamang sirs. I am very much thankful to the Gut Microbiota Probiotic Science Foundation for selecting our work as the first prize in the poster sessions. And I'm also very much thankful and grateful to Professor Ganguly sir, Srivastav sir, and other dignitaries present here, students, friends. Our work on influence of supplementation of lactobacillus and Saccharomyces means the combination of lactobacillus and yeast on growth performance, blood profile, cholesterol contents, and metagenomic analysis of the broiler chickens. Basically, our purpose is to the previous day's discussion on AMR, antimicrobial resistance. So our main motto was to produce the antibiotic-free meat and egg production in the broiler chickens. So we have removed the antibiotics or the growth promoter as well as the commercial probiotics given to the broiler chicks. So all of you know that day by day the antibiotic resistance are the great concern for all over the world. Even in our state, we are funded by our state government to see the antimicrobial resistance in milk and milk products, and we are getting MDR, multi-drug resistance pathogens in the milk and milk products, mainly fermented milks prepared in the domestic level. So it's a great concern for all of us to see into the AMR. So on the basis of that, we have supplemented these two organisms. One is Lactobacillus fermentum, and the one is the Saccharomyces cerevisiae. So for this study, the experimental design is the 96 one-day-old broiler chickens were purchased from the Venki's lab, and the chickens were wing banded and randomly assigned to 4 different groups. The groups are, first one is the control. Control means it is a commercially available feed has been given to the chicks, but T2, T3, and T4 is the test group where we have not given any kind of probiotics or immunomodulator or growth promoters. And instead of that, in T2, we have given only the Lactobacillus fermentum. For T3, we have given the yeast only. And in T4, it is a combination of Lactobacillus as well as the yeast. And the study was for 42 days. And we have feeded the chicks from 8th day to 15th day on a daily basis in these 3 test groups. And the cultures which we have selected, they are being tested that they are safe by doing the test like gram staining catalase test, indole test, urease test, phenylalanine test, hemolysis test, gelatin hydrolysis test, mucin degradation, as well as the biogenic amine productions. So for all these tests, these 2 organisms showed the negative results. And in the pigeon study, these 2 cultures were considered, and during the entire study, the higher body weight was observed among the lactobacillus fed broiler groups as compared to the control group. And HDL cholesterol was even was found to be higher in the treatment group T2, where only the lactobacillus itself is the supplemented in the food. And the results obtained indicate that the potential use of lactobacillus as well as the yeast as a dietary feed supplements for the alternatives to the antibiotics as a growth promoter in the poultry feeds. We have also tested the anti-inflammatory activity of these 2 organisms against the TNF alpha, IL-6, IL-1 beta. And also we have checked the nitric oxide production as well as the cell viability by raw macrophage cell line 264.7. And we got very good results in the yeast. If you see the TNF alpha, IL-6 and IL-1 beta, the single yeast can reduce the TNF alpha, IL-6 and IL-1 beta when we are treated with the LPS. So we got very good promising results in the anti-inflammatory activity of these 2 organisms. And this study, basically, I already said that there are 4 different groups we have taken. And if you compare with the body weight, the T2, where the only lactobacillus fermentum was given, they are having a good amount of gain good amount of body weight, even feed consumption ratio also. So the average body weight of these chicks or the birds fed with the lactobacillus strain KG4 was found to be numerically highest as compared to the T1 is the control group, and T3 group fed with the yeast itself, and T4 is the combination of yeast plus lactobacillus. We have also checked the hematological parameters, mainly WBC, RBC, hemoglobin counts, and platelet. So most of the cases, the results are at par with the control. In some of the cases, you can see that the hemoglobin level, even platelet, T2 has given a good amount of hemoglobin level. Even in the T3, where only yeast has been given, the platelet count is more compared to the control. In the cholesterol profiles or the lipid profile analysis, HDL cholesterol has been increased in the T2, where the only lactobacillus has been given.
Dr. Subrata, please go to the conclusion.
This is the histopathological analysis. If we compare with the control group, these are three different organs: intestine, liver, and heart. And there is no any necrosis or any evidence of hepatic necrosis was found in all these organs. They are all compact and normal as compared to the control one. We've also analyzed the fecal samples of these different groups, and we got very good amount of lactobacillus counts in the T2 group because we have treated with the T2. We've also done the metagenomic analysis, and we have seen that Akkermansia genus was highly abundant in the T2 group, as well as comparatively more abundant than the other groups. And mostly there was no significant difference was observed when we compare the control or the T1. The metagenomic analysis, we have also done the alpha diversity measures also. So the results were non-significant because our main aim was to compare with the control, because control has given the immunomodulator or growth promoters where we have not given. So the conclusion of this study is that the oral feeding of the Lactobacillus fermentum and Saccharomyces cerevisiae individually or in combination alone with a basal diet without adding immunomodulator to the broiler chickens improved the growth performance during the 42-day studies. Further, the gain in body weight in Lactobacillus was higher, and good cholesterol was found also in the Lactobacillus, but in combination has given a more good results, and we have also published this in the Journal of the American College of Nutrition recently. We are also acknowledging our institute as well as the gut microbiota and the DBT. So at the just take home message that it is very favorite line for me: say no to drugs, only yes to belly bugs. Thank you all for listening me.
Thank you, Dr. Subrata. It's for very nice work. We can take only one question, quick question. If there is any. Yes, please. Quickly, please give the mic. The backside. Backside. See.
Thank you. Very nice presentation.
Thank you.
Just wanted to understand, do you have already performed or have plans to perform any challenge study with this probiotic supplementation?
No, we did not do any challenge studies. Only we have removed the immunomodulator or the growth promoter, because you know that in the chicks at the very beginning they give some kind of growth promoters like antibiotics so that their body will gain in a very short span of time, and these organisms are getting antimicrobial resistance. So our main target is to remove those immunomodulator or growth promoter, and we have supplemented our own two lactobacillus. One is lactobacillus, another one is yeast. We did not do any challenge with them. Thank you.
Thank you, Dr. Subrata. Rest we can interact with him during the lunch time. I will now request my colleague to introduce the next speaker. Thank you so much.
The next speaker is Mr. Basava Prabhu, HN. He is a PhD scholar of NDRI Karnal.
Yeah.
I think online.
Dr. Prabhu, are you there?
Yes, sir. I'm presenting. Am I audible, sir?
Yes, very clear.
I'm just sharing my screen. Yeah. Very good afternoon to. Hello. Yeah. Am I audible?
Yeah.
Yeah. Very good afternoon to one and all present here. I'm Basava Prabhu Ethan, and I'm here to present my Young Investigator Award presentation on the topic Integrated Preclinical Safety and Efficacy Analysis of Potential Probiotic Lactobacillus fermentum NCDC 400, a computationally guided in vitro and in vivo investigation. As we know that probiotics are the beneficial microorganisms that provides health benefits to the host. And when we look into the classical requirements of the probiotics, what we need is, although we have several classical requirements, I focus on the safety of any probiotic strain for the human consumption. For that, because we have several reports stating certain adverse effects of probiotics, either theoretical effects or or the adverse effect in case of immunocompromised individuals, such as septicemia, bloodstream infections, adverse metabolite production, drug resistance, infectivity, and virulence factors. All these factors makes us to evaluate the safety before we translate any probiotic strain into clinical aspects. As well as for that reasons, we have our own Indian guidelines to study the safety of probiotics. For that is in ICMR DBT guidelines. Based on these guidelines, we have framed. We thought of investigating the safety of this strain by formulating three objectives, that is assessment of Limosil lactobacillus 400 and NCDC 400 for safety aspect using in vitro method. And second one will be the toxicological evaluation of NCDC 400 in the murine model. And third will be studying the safety and efficacy of the NCDC 400 in an immunocompromised murine model. Coming to the methodology, we have amalgamated in silico, in vitro, and cell culture approaches to evaluate the safety. At the for foremost thing, since we had the whole genome sequence of the strain, we have used bioinformatic tools and databases to study the safety. Followed by we have done an array of in vitro safety tests. Followed by we have done oral toxicity tests in healthy animals as well as in case of immunocompromised animals. And then we have compiled our results, and these are the results that I'm going to discuss. As we can see here, the test strain was not showing hemolytic activity, and it was coagulase negative, DNA is negative, urease negative, and it was also negative for phenylalanine degradation, indole test negative, as well as gelatinase test negative. And the test strain was not aggregating the platelet, and also it was producing comparatively less amount of ammonia compared to certain pathogenic strains, and also it was sensitive to the human serum, and it was producing a majority of the L-lactic acid, and by this we can get to know that the possibility of D-lactic acid mediated acidosis is comparatively less in the strain. And then we have done the mucin degradation assay, and it was found that the strain is not going to degrade the mucin, and it was not producing any of the tested biogenic amines. And also it was not producing any harmful intestinal enzymes like beta-glucosidase and beta-glucuronidase, and the test strain was safe to the CACO2 cells, and also it was adhering superior adhesion to the CACO2 cells as well as the porcine mucin, and also it was not breaching the gut enterocytes as we studied in the CACO2 cells. And also we have profiled the antibiotic resistance and of this strain, and we found that the test strain was following the criteria which was given by EFSA, and it was sensitive to all the antibiotics given by EFSA, and also it was not having any resistance or virulence genes which we have tested as through the PCR assays. And also through the whole genome analysis, we got to know that this bacterium was having the vancomycin resistance gene, which is an intrinsic resistance, and it is not going to cause the horizontal gene transfer episodes. And also we have found a series of resistance genes in this strain, but all of these are belonging to the aminoglycoside antibiotics, again, which is an intrinsic property of any lactic acid bacteria. And the strain was comparatively stable, had the stable genome because of incomplete prophase sequences and also one CRISPR sequence, and the strain did not carry any of the virulence factors, and therefore it was considered as and predicted as non-pathogenic to humans. And also it was not having any of the pathways that can cause that can produce the metabolites that cause the platelet aggregation, and it was not having any pathways to produce the biogenic amines, and also it was not having any origin of transfer regions that can able to transfer from one to another. And then this is the animal study which was done. Since we have done acute, subacute, and subchronic toxicity study, I am presenting the data of subchronic toxicity study for 90 days. As the probiotic supplementation at low dose and high dose. Low dose was 10^8 cells, and high dose was 10^10 cells per mouse per day. For 90 days, there was no significant variations in the general health status of the animals, and also there was no changes in the physical appearance. And when we look into grass pathology or necropsy, there was no any significant variations in the organ morphologies as well. Further, there was no changes in the organ index across the group, and also the hematological parameters did not vary significantly across the group, as well as the clinical biochemistry parameters also did not vary across the group. And also when we looked into the gut health indices, like fecal, all the gut health parameters were significantly improved after probiotic supplementation, either at low dose and high dose. And also there was no any bacterial translocation to the extraintestinal organs, including blood, liver, kidney, spleen, and suggesting that probable sepsis or bloodstream infection due to by the strain is absolutely nil. And also there was no any, upon histopathological examination, we got to know that there was no any tissue or test-strain related microscopic changes in the tissues. And this is a safety study, last study that we have done in the immunocompromised mice model. We have used cyclophosphamide to create the immunocompromised model. As we can see here, in case of the model control, that is the mice group that received only cyclophosphamide, there was a drastic reduction in the body weight. But such drastic reduction in the body weight, feed intake, and water consumption was significantly lowered in case of the probiotic administration, suggesting that it might have helped in the feed conversion ratios. And also there was significant improvement in the organ index was seen in the probiotic supplementation groups. As we can further see here, when we did the CBC, complete blood count analysis, we found that WBC content was significantly improved, although not significant, but there was an improvement in the WBC content, suggesting its immunomodulatory effect. And also there was an improvement in the clinical biochemistry parameters in the probiotic supplemented group. When we looked into the immunological parameters, there was an increase in the IgA, IgG, IL-6, IL-10, TGF-beta, TNF-alpha, MCP-1, and nitric oxide content, suggesting that the immunomodulatory activity of this probiotic strain, which was augmented after the administration of the strain, and the macrophage activity also significantly improved. That is, peritoneal murine macrophage activity increased after probiotic supplementation. And also the T cell and B cell activation, the proliferation, the splenic cell activation was also improved significantly in case of low dose and high dose. And the oxidative markers like catalase, SOD, and GPX2, that is glutathione peroxidase, increased significantly, suggesting the antioxidant property of this test strain. And also the strain was non-genotoxic as we tested through the micronucleus assay and in vivo chromosome.
Please come to the conclusion.
Sir, definitely. Also the gut parameters increased significantly, and also there was no microscopic changes that happened in case of the other tissues. At last, we have done the gut metagenome analysis of the fecal microbiome, and there was an improvement in the diversity and richness in the probiotic, the microbiome in the probiotic supplemented group, and there was a reduction in the pathobiome, increase in the beneficial bacteria and short-term producing bacteria that was seen in this study. At the conclusion, I would like to draw the conclusion that the strain was safe and it was non-pathogenic, and a dose of 10^10 CFU per mouse per day can be considered as no observable effect level. And also, the strain was immunomodulatory and it was well tolerated in the immunocompromised mice as well. So, by these observation, I can put in the way that the future prospect will be like. This study should facilitate further human trials, like focusing on phase 1 and phase 2, phase 3 clinical trials. At last, I would like to thank everybody. Thank you.
Any question? Only one question.
Sir.
Any question? If no question, then thank you very much. We'll go to the next.
Thank you, sir.
The next speaker is Ms. Baishali Dasria. She's also from NDRI Karnal.
Eight minutes, huh, Vishali?
Can I start, sir? Good afternoon to all attendees of the 13 India Probiotic Symposium. Imagine waking up one day unable to remember your name, your family, and the most precious moment of your life. That is the reality of the millions of the people around the world who are suffering from the Alzheimer disease. So Alzheimer disease is the devastating condition that slowly robs the individuals, their movements, and the cognitive ability. So what if there is a way that to prevent or slow down the onset of the Alzheimer physiology, where that is where the probiotics come in. So here I am Vaishali Dasaria, PhD scholar at the ICR NDRI Karnal, going to present on the topic of gamma producing lactobacilli exhibited the anti-Alzheimer potential in the animal model. As stated by the Fringe theory, the gut bacteria not only modulate the physiological homeostasis condition, but it also maintain the mental health. That is, they profoundly shape our thinking and behavior. So from that, I have collected the lactobacillus cultures from the NCDC and DRI Karnal, and I have screened for the neuroprotectants such as the GABA, vitamin B12, Soch and fatty acid, and antioxidant potential. So from that screening, I have got one cultures, lactobacillus fermentum NCTZ 701, that's showing the higher GABA producing vitamin B12 and having the better antioxidant potential with the Soch and fatty acid, especially butyrate, which are mainly responsible for the decreasing the amyloid plaque deposition. As we know, the Alzheimer is the condition in which there is the increasing in the amyloid plaque deposition, alteration of neurotransmitter level, and also increasing the oxidative stress. The Alzheimer disease also caused due to the dysbiosis of the gut microbiota. So the selected cultures NCTZ 701 is used for the further study like animal model. So we have developed the animal model of Alzheimer by using the induction of aluminum chloride and D-galactose at the different dose, lower, medium, and high dose. So I have selected the low dose of aluminum chloride and D-galactose for the Alzheimer model for the duration of the 7 days. I have found that in the induction of the aluminum chloride and D-galactose on the 7 days, I have found there is a decrease in the cognitive decline. I have checked by using the open maze field, sucrose preference test, and the novel object recognition test. Then again, I have found the alteration of the neurotransmitter level also. And also, I have found the amyloid plaque deposition in the brain by using the histopathological analysis. So again, I have divided the albino vista rat into 6 group, and last 3 group, 4, 5, 6 group have given the probiotic in different 3 conditions like a co-administrative, prophylactic, and the therapeutic. So I got result that there is also improving the condition of the cognitive ability by using the open field method. I have found there is increasing ambulance time and decreasing the resting time, which are mainly responsible, which are higher resting time, higher in the Alzheimer disease. There is also found that there is improving the condition of the blood profile. Like lactobacillus treated group found that there is increase in hemoglobin, RBC, increase the lymphocyte level, and increasing the platelet distribution with cell. In contrast, there is the decrease in the platelet volume. I have also checked the vitamin B12 in the plasma of the rat. I have found that there is an increase in vitamin B12 in all remaining means 3 group of the probiotic treated group as compared to the Alzheimer group. I have also checked the amyloid plaque deposition. I have found there is a recovery of the means reduction of the amyloid plaque deposition in all 3 group. I have also checked the lymphocyte infiltrated cell in the brain tissue. I have found there is a decrease in the infiltration of lymphocytes in the brain of lactobacillus treated group. So we check the amyloid plaque deposition. There it is found that the decrease in amyloid plaque deposition as compared to the Alzheimer group. As talking about the LPS, there is found that there is a reduction of the LPS content of the brain in lactobacillus treated group. We have also checked the inflammatory parameters. We have found that there is a reduction of the pro-inflammatory cytokines such as interleukin IL-6 and TNF-alpha, and increasing in the IL-10, which are the anti-inflammatory cytokines. We have also checked the short chain fatty acid by using the GLC of the fecal sample. We found that there is the increase in the short chain fatty acid in the lactobacillus treated group. So in group 2, there is more production of the propionate and acetate. It may be due to the decrease in the Lycospiraceae firmicutes and increase in the level of bacteroids. We also found that in group 3, which is a positive control treated with the memantine drug, they have found that there is abundance of firmicutes, and they also found that abundance of firmicutes correlate with the butyrate production. In treatment group 4, 5, and 6, we found that there is an increase in the short chain fatty acid, total short chain fatty acid, and especially butyrate. So it is correlated with the 16S sequencing. Found that there is the increase in abundance of the Asclerospira, lactobacillus, and Rhinococcus. As talking about the antioxidant activity, we have found that better antioxidant potential of the Lactobacillus fermentum 701 in treatment group. So we have also done the 16 year sequencing.
To conclusion, Vaishali.
Okay, sir. As we know that Shannon index and observed OTU mainly responsible for the richness and the diversity, we have found the increase in the diversity. And we also found that there is the modulation of gut microbiota and decreasing in the pathogenic bacteria which are responsible for Alzheimer, like a Tannerud's and the Coprococcus. So we also found that there is enhancement of the tight junction gene and also decrease in the inflammatory gene. So this is the overview of my work. So finally, I conclude that Lactobacillus fermentum 701 significantly elevates the pathophysiology of Alzheimer's disease, and future clinical safety trials can be performed to validate the probiotic-based or biofortified milk-based therapeutic interventions for the Alzheimer's disease. Thank you.
Thank you very much, Vaishali ji.
Thank you, sir.
I think because of positive time, we can interact with all these three youngsters during the lunch time. Do we have the fourth one? We had third prize, Manisha Sethi, also from Institute of Life Sciences. Manisha, are you there online?
Yes, sir.
Okay. Then.
Yes, sir. I'm there. Sir, am I audible?
Yes, you are quite audible. I request you to finish your talk in 7 minutes, please, because we are running behind the schedule. So, Manisha, please start your presentation.
Yes, sir. Thank you, sir. Good afternoon, everyone. I'm Manisha, working as SRF at Institute of Life Science under the guidance of Dr. Santibhushan Senapati. The title of my work is Lactobacillus salivarius F14, originated from tribes of Odisha as a potential probiotic. Before going to the work, first of all, I want to thank all the organizing committee members and the judges for selecting my work for this esteemed award and giving me this opportunity to present my work here. Coming to the introduction.
It is better you directly come to results, no? Because don't waste time in other things. Because.
Okay, sir. Sir, I just want to say that about 4 years back, our institute initiated a flagship project on tribal health and nutrition, in which our lab took the responsibility to isolate and characterize potential probiotic from the stool sample of the tribal people with an overall goal to formulate some personalized or community-based probiotic for the tribal people. And for that, here the cartoon shows the strategy. And so far, we have isolated around 25 probiotic strains. And today I am showing the work related to only one of the isolate, that is F14. So these are the results. We have from the initial phenotypic characterization, and we confirmed it is a lactobacillus species, gram-positive rod-shaped one. And we also performed a set of experiments as per the ICMR DBD guidelines to check its probiotic attributes, and it passed all the tests to be called as a probiotic. Then in our next approach, we try to check its anti-salmonella efficacy by in vitro and in vivo experiment. Our in vitro data also supported that it is showing good antimicrobial property against the Salmonella pathogen. Then we also performed whole genome sequencing for its strain level identification, we suggested that it is Legionella lactobacillus strain having 137 unique genes. And from the metabolomic analysis also, we found that its CFS is able to contain so many important metabolites having immunomodulatory antimicrobial properties. Then in the end, we did in vivo study to evaluate its, this is my last slide, sir. In vivo study to evaluate its anti-salmonella property. Here the figure A is showing the experimental strategy. And from the result, we observed that this F14 probiotic is able to increase the survivability and restore the body weight and normal tissue architecture, as well as it is reducing the pathogen load in the intestinal cecum and liver, as evident from the CFU data and live imaging. And as well as from the ELISA data also, it is indicating that in both serum and intestinal flux, it is it is trying to normalize the IL-10 level, supporting that it is trying to create an immune balance. So all this data indicate that this F14 probiotic has the potential to reduce the pathogenesis of the Salmonella typhimurium in mice. Parallel to this, we also thought to do a preliminary study to check its immune modulatory status, and we found it is able to increase the Treg population in the spleen, as well as it is able to increase the mRNA level of IL-10, MRC1, and TNF-alpha in the paired species. However, to claim this immunomodulatory property, we are planning to further study it in detail. So the overall work can be summarized as F14 bacteria is the lactobacillus species having probiotic properties, and the whole genome sequencing confirmed it is Legionella lactobacillus strain having 137 unique genes, and both in vitro and in vivo study suggested that it has the potency to reduce the pathogenesis of Salmonella. I want to thank our present and former director, and I want to thank my PI, Dr. Santi Bhushan Senapati, for his constant support and guidance, all the lab members and collaborators, all the funding agency, volunteers, and lastly the mice who sacrificed their life for the study. Thank you all.
Thank you very much, Manisha. It's a very nice piece of work, but because of positive time, we are not able to discuss it. Anyway, at the moment, I would like to conclude the session. No doubt there were 4 very, very interesting presentations. And these are the young persons who's going to be the future of Indian probiotic research. We have 20 more in the audience, and I request everybody to look at the posters which are presented there. I will request my colleague, Dr. Tamang, to say a few words.
I don't have to say anything, but on behalf of the gut foundations, under the able leadership of Professor Ganguly, the main objective of our gut foundation is to ignite, motivate the young researchers in India, especially in the field of the probiotics and related fields. This is what we have been doing for last 13 years, and continuously every year we organize this, and we have a very rigorous type of the screening, and we invite the abstract from the different institutes, universities all over India. Then there is a rigorous screening process. We have the panel of the experts, the internally and externally. Then after that, this time we have more than 45 applications, the abstract. Then out of that, we selected 20. And out of that 20 also, we have the jury of the panels from the internationally recognized scientists from the abroad. They screened, and this four of them have been selected. So we congratulate all these 4 and as well as the 20 also. So this is the thing. Then I think you can go to our website. We always upload the latest publications, whatever is going in the field of the probiotics and all. please go to that website and our newsletter also. Thank you.
Thank you to our dignitaries for diligently conducting the session. I would now be requesting the humble presence of Mrs. Jatana to kindly join us here on stage and felicitate our chairpersons herewith. Can we welcome Mrs. Jatana with a round of applause? Thank you, ma'am, for coming in. I would request Mrs. Jatana to please come and felicitate our chairperson here, Professor J.B. Prajapati, sir. Absolute honor. So please accept this token of appreciation from our end. It is a delight to have you here with us. I would also thank you, Mrs. Jatana. I would also request the humble presence of Mrs. Veena Seth to kindly join us here on stage as well. Veenu Seth, ma'am, to please join us here on stage. Mrs. Veenu Seth, can we welcome the lady with a round of applause, please? Most welcome, ma'am. Thank you for coming in. Please come. Most welcome here on stage, ma'am. I would request Mrs. Seth as well to be present here on stage with our dignitaries. These are our venerated guests, and it is a delight to have their humble presence here with. So thank you so much. Thank you to the ladies and thank you to our chairpersons as well. It's okay. It's okay. Thank you. That's okay. All right. We now proceed forward to our next segment of the event, which is the panel discussion on the topic probiotics in combating non-communicable diseases. For this session and the most exciting part of the symposium, which is the panel, I would be requesting the humble presence of Dr. B. Sasikaran to moderate the panel. Dr. Sasikaran is the former director of National Institute of Nutrition, ICMR, Hyderabad. He is a pathologist by training and carried out research in the area of nutrition, food safety, toxicology for 30 years.
Yeah. Now we are behind time, so there's not much time for niceties. Now, first of all, my apologies to all the speakers in the previous session for having rushed them through, which I think was not very fair for those young people. Now we have barely half an hour left. Now I invite Mohan, you're already here and you've been here for quite some time on and off. I'm sorry to have kept you waiting. And Dr. Namita Chandyok, Dr. KP Kochar, Professor Seema Puri, Professor Ajay Balla. I request all of you to kindly come onto the dais and take your place, please. Now, this is the final session with the least amount of time, but the most exciting areas of clinical research, and it covers the whole spectrum of non-communicable diseases and the role of use of probiotic and what is the evidence that lies with us. And do we really have enough information on these to recommend or utilize these? And I'll first request, I think I'll ask the questions so that you can either answer it without presentation or make a brief presentation, not more than 5 minutes. So I start with Dr. Mohan. Mohan, you can hear me? So Dr. V. Mohan.
I can hear you.
Yeah.
I just wanted to check whether you can see my slides because I was told you can't see my slide. Is it visible now?
Yes.
It's visible. Okay.
Now, Dr. V. Mohan is the chairman of the Dr. Mohan's Diabetes Specialty Center and Madras Diabetes Research Foundation, Chennai. He's one of the topmost diabetologists in the country. He has more than 400 and odd papers in the area of diabetes. And I think he's the best authority whom we can ask this question. Dr. Mohan.
Yes.
What is the role of probiotics in diabetes as far as the literature goes? And what is the clinical situation in your practice? Do you use probiotics? And based on the information research, do we have still lots of gaps in this research on the use of probiotics in diabetes? Your time is 5 minutes, sir.
Thank you, Dr. Shikharan. Let me thank Dr. Ganguly and Neerja and yourself for this session. What I thought I'll do is I'll give you a quick review of the literature. Actually, you can see most of these references are quite new. So a few years ago, when we had the meeting in person before COVID hit us, we really didn't have any evidence at all that probiotics are useful in the treatment of diabetes. This has changed quite a bit. So I thought I'll just show you some meta-analysis of recent studies and systematic reviews. Here is one from New Jersey, which shows it's a randomized clinical trial probiotics, which shows that multi-strain probiotics that contain 7 million to 100 billion colonies seem to be efficacious in improving glycemic control in adults with type 2 diabetes. So this is a very powerful statement. Here's another one from Hungary, published in Scientific Reports. And this group says that our data clearly suggests that probiotics could be a supplementary therapeutic approach in those with type 2 diabetes to improve dyslipidemia and to promote better metabolic control. And according to our analysis, probiotic supplementation is beneficial in type 2 diabetes. Here's another one from Brazil, published in 2021, which looks at probiotics, prebiotics, and symbiotics. 38 articles were included after literature search. It's a systematic review and meta-analysis. And they conclude that overall, the use of prebiotics, probiotics, and symbiotics reduced A1C levels, but did not reach the level of significance, and they had no effect on LDL cholesterol. However, they did decrease fasting glucose and total cholesterol. Here is another one from Thailand and US, published together in Advances in Nutrition, which looks at, again, systematic review and meta-analysis. And they conclude that probiotics may have a glucose lowering effect in type 2 diabetes. And this effect seems to be stronger in participants with poorly controlled diabetes and those who are not on insulin. So we're getting some hints where it may be useful. What about the role in pre-diabetes? Now, in pre-diabetes, this particular study from China, published in 2021, says that there may be some partial benefit, but as of now, the evidence is insufficient to promote it in the treatment of pre-diabetes. What about type 1 diabetes? Again, the study published quite recently, 2022, says that there are several theoretically possible mechanisms in type 1, including modulation of gut microbiota, the regulation of inflammatory-related cytokines, and production of short-chain fatty acids, and so on. But they recommend that more studies are carried out. So clearly in pre-diabetes and type 1 diabetes, the evidence is emerging, which we did not even have for type 2 diabetes. But in type 2 diabetes, at least 3 or 4 reviews suggesting that it is useful. Now, this last but one slide shows you the natural history of diabetes. And the question will come, when do you use prebiotics or probiotics? Do you use them in those with normal glucose tolerance, a stage of primordial prevention? Do you use them for primary prevention, pre-diabetes? Do you use them in clinical diabetes stage to delay the onset of complications? Or do you use them after the complications have set in tertiary prevention? And this last slide, I like this because it kind of puts it into perspective that there are several mechanisms, first of all. And the hypothesis is that probiotic intervention can help to decrease the incidence of diabetes, which means in pre-diabetes. It can reverse the extension and therefore slow down the progression. That means the development of complications can be slowed down. And in those who already have some complications, maybe it will limit the damage. Of course, this is more theoretical, and the studies I presented earlier are the ones which show some evidence. So to answer your question, do I use it in practice? Of course, I use it in practice, particularly in those who are above 60 years of age, because I feel that their gut health definitely improves by the use of probiotics. I mentioned this earlier. Those who have constipation and other GI-related problems, I don't hesitate to use probiotics. Of course, if they use antibiotic use, I routinely use it. And in any other situation where there are any symptoms, I use it. I don't use it in the treatment of diabetes as such. Okay, your sugar's not come down, add probiotics and see, because I think the reduction in A1C is 0.2, 0.3. And so far, no guidelines approve it as a treatment for diabetes along with metformin or along with any other drugs. But having said that, there are certain drugs which can adversely affect the gut microbiota, and therefore possibly the use of these will start getting recommended in the future. So I'll stop there. Thank you.
Thank you, Dr. Mohan. That was an excellent summary. And please don't go away because the audience have some questions for you, sir. I invite my colleague in ICMR and former senior DG of ICMR, Dr. Namita Chandyok. And she has a lot of experience in the area of work in polycystic ovary syndrome, is again another non-communicable disease sort of leading up to insulin resistance and several complications. Dr. Namita, would you like to make a small presentation or? Yeah, please go ahead. Yeah. If possible by 5 minutes. Yeah. Yeah.
So good afternoon, and thank you, Dr. Ganguly and Neerja, for inviting me and giving me the opportunity to speak on a topic which I think is increasingly going to be very important, because especially polycystic ovarian syndrome, we are seeing so much of it. It's such a complex, heterogeneous, and a common disorder which is affecting such large number of women. You talk to any gynecologist and she's going to tell you that every 1 out of 3 or 4 patients which come to her have symptoms of polycystic ovary syndrome. And the worrying part is that not only does it have problems which are related to the reproductive tract, but it also has very long-term side effects which continue beyond the reproductive age group. And these are mainly associated with all the metabolic dysregulation which occurs. And these are really the ones which are of concern. So it becomes very important for us to understand what the etiology and pathogenesis of this disease is, so that we can facilitate development of novel therapies. And so the etiology of PCOS is really not clear as yet, and it is thought that it is, it is seen more in people who have a genetic predisposition, along with the effect of environmental toxins and an interplay of diet and lifestyle. And this leads to the hormonal and the metabolic imbalance and gives us the features of PCOS. But increasingly, gut dysbiosis has now been seen as another factor which has been associated with PCOS. And that is really what we're going to be discussing today. So gut microbiota, I'm not going to go into it because, you all know what a complex ecosystem it is and how it affects the body in so many ways. But if you see the gut microbiota changes in PCOS, there are significant differences in the composition and also in the diversity and also what we see in the relative abundance of certain bacterial taxa. But there are inconsistencies in the studies, and while some studies show these changes, others do not. And so really these are, more work needs to be done into it. And more studies which are going to see what are the mechanistic changes to understand how these microbes really influence the development and the pathogenesis of PCOS. The dogma theory, which you are all perhaps familiar with, was, proposed many years back. And this hypothesis, which really talks of how this is a drug-induced dysbiosis, which leads to increased permeability, and then the endotoxins which are released, leading to immune system activity, which disrupts the insulin receptors and at the elevated serum insulin levels, and which stimulates the production of androgens, interferes with the follicle formation, and leads to the establishment of PCOS. But the gut microbiota, the dogma theory, which associates diet with the changes in the PCOS, lays a lot of emphasis on obesity and also on the insulin resistance. And we know that, we can have PCOS also in those who are lean, and in not all cases of PCOS is insulin resistance seen. Besides that, also in the prevalence of PCOS is more or less the same worldwide, even though there are differences in diet. And animal models of PCOS have been created which are independent of diet. So this hypothesis really is not able to explain why there are changes in the why there is this association between PCOS and the gut dysbiosis. Now, the other hypothesis is of how the hyperandrogenism drives the gut dysbiosis in PCOS, and they have, even though there are no studies which are being able to tell the causality, because you cannot do that in humans, but there are multiple studies which are reported which show an association between the hyperandrogenism and the changes in the gut microbiome. And as the testosterone levels are increased, you can see that there is alterations in the diversity and in the composition of the gut. And and this was also shown more in a study where letrozole was used to induce hyperandrogenism and PCOS in rodents. And those which are the PCOS-like symptoms. And when they saw the gut microbiome, there was gut dysbiosis, which was because of the hyperandrogenism, and this kind of gave some kind of an association between the PCOS and the gut, the hyperandrogenism and the gut dysbiosis. So between the hyperandrogenism, the dysbiosis of the gut, and the metabolic dysfunction, all these factors, there is some interconnectedness. But how each of these factors alters the other two, that is still not clear, and further research on this needs to be done. So both these hypotheses, which I mentioned earlier, they are interrelated to each other and also to the metabolites which are released, and these are altered in PCOS, and also they have an effect on the gut microbiota. So we know that the gut microbes, they metabolize the substrates, and the metabolites which are released are either then are back in the gut, or they go back into the host metabolic tissues. To the host metabolic tissues like the skeletal muscle, the liver, the adipose tissue, and to the ovaries, which all are affected. These tissues are affected in PCOS, and the metabolites which are altered are the secondary bile acids, the short chain fatty acids, and the trimethylamine. Even in the host metabolic tissues, all these tissues, all the metabolites which come out and the ones which are altered, they have a direct effect on the gut and can lead to dysbiosis. In the host reproductive axis, where the steroids are made, the androgens, the high testosterone level, again has an effect on the gut microbiota, and there is independent crosstalk between the host reproductive axis and the host metabolic tissues and the metabolites, which then further act on it and can make a change to the gut microbiota. And so, knowing this plausible mechanism of action, what can be done? So perhaps restoring the gut microbiota through prebiotics, probiotics, and FMT would have an effect and could be used to treat or to prevent PCOS. And, with this, probiotics have been used. And I think a lot of studies, again, on probiotics have been carried out, and a lot of interest has been generated because of this. And if you see huge number of studies, I mean, the interest was so wide that in the last 5 years, I came across 18 RCTs on probiotics, and has shown that there is effect on all the glycemic control and also on the hormonal level. But a meta-analysis showed that it had some effect on the hormonal control, like there was reduction in sex hormone binding globulin and the free androgen index.
Can you conclude, Dr. Amita?
Yes. And but not in all. And so I think where probiotics are concerned, they could be a useful therapeutic adjunct to the dietary and lifestyle modifications. As of now, there are no guidelines for it, and whether they are being used in clinical practice, I'm really not aware, and I really don't think so. But the important thing is that the actions and characteristics of the probiotics are strain specific, and those actions cannot be extrapolated to others. So really further studies are really needed to really really know what is the strain-specific efficacy, what should be the dose, what should be the duration, and whether what are the health benefits of the clinical outcomes in the PCOS. The same for prebiotics. Very few studies, and most of the studies which have been done are as a symbiotic, and only there was this one release study which did show that there was a reduced in both the hormonal and in the biochemical parameters. For symbiotics, one study which did show, which was a double-blind placebo-controlled RCT, we did show that it was of benefit. For the fetal microbiota transplantation, there have been no human studies. There have only been animal studies, mostly in rodents. But it is, I think it could be a viable cutting-edge treatment in future. But at the moment, further research is required before, we can move forward towards this. So I would like to end by saying that PCOS is associated with biodiversity and changes in the bacterial taxa. And but further research is needed for us to really know what are the mechanisms. So we really need to do mechanistic studies to really understand what is the mechanism which is underlying the present association which we are seeing. And the prebiotic, probiotic, and symbiotic supplementation and FMT have shown to have beneficial effects, but I think the really further research is required before, they could really be used as treatments for the PCOS therapy or for the prevention. Thank you.
Thank you, Dr. Namita. Dr. Mohan, you think if there are probiotics which can reduce insulin resistance, they will also have a spin-off benefit in bringing down PCOS? Maybe Mohan can answer it later on. Now, another problem which really is for which we have very few solutions are the neurodegenerative diseases, which is the biggest problem with aging. And now to discuss the role of probiotics in this area, we have Dr. K.P. Kocher, head of the Department of Physiology from the All India Institute of Medical Sciences. Madam, can you just tell what is the recent research in the area of the gut-brain axis associated with probiotics and whether it is likely to really benefit the neurodegenerative problems which comes along with aging? I think you have a couple of slides. Would you like to share? Sure. Good.
No problem. I can without wasting time.
Yeah. Unless, of course, you want to wait for some time.
No. I don't want to wait. Everybody is. Sir, if you allow, I'll just, from whatever I've heard till now, I just wanted to say that we do cognitive neurophysiology and nutrition, and three areas. One is probiotics, one is coarse cereals, and the third is use of spices. These are the three domains of nutrition which are very specific to a traditional Indian lifestyle. And obviously, students here will agree that when we do a research, we follow a reductionist approach. But actually, when we do translation and applicability, then it becomes more of a systems-based approach, just like Professor Chairman Sir was telling us that what will happen if the insulin sensitivity is improved after probiotics, and it will help in the PCOS. So we are doing a phase 3 clinical trial on Parkinson's disease with fluctuating symptoms. These patients have had dopamine for quite some time. They are in the senior aged group, and we are doing studies on cognition. We are measuring their complete autonomic profile. Yes, sir. Is it on? Can I come there? And along with it, we are doing the translational model also. So one PhD student is working on human studies and studying cognition in 4 domains of attention, executive decision making, memory, And the fourth most important, that is motor planning, because whatever comes to us from the environment, the end result is outcome is a motor response or a programming. Along with it, another student is working on a 6-hydroxydopamine rat model. And can we quickly run the slides, or should I come there?
He can help you.
Quickly, yeah. So this was the study design. Next one, please. So we are doing both, as Madam Chandok had referred to the mechanistics. Next slide, please. Yeah. So this was our theme for the humans, where we did measurements, quantitative also, as well as neuropsychological questionnaires. And we did a bold imaging or non-invasive neuromapping of the brain by sending in infrared rays. And once it is emitted, and the principle was the hemoglobin oxy versus deoxyhemoglobin has a differential wavelength, and by Beer-Lambert's law, we equate it with the neuronal activation. And in the rat model also, we did a lot of studies, right from the patch clamp studies in the striatal neurons to doing neurobehavioral analysis, and also doing the memory tasks and the maze tasks, and also evidencing the histology of the brain as well as the gut, and doing some studies on metagenomic 16S RNA sequencing. Next, please. So these are some details, whatever we are doing. Next, please. Yeah, so these are some pictures which show. The panel top left shows a study which we have pioneered in India, that is called Beer-Shafts potentials or movement-related cortical potentials. They are a very good tool to evidence changes in motor neuroscience, motor planning and motor execution when we correlate synchronously the EEG as well as the EMG. So before 1500 to 1200 milliseconds before. So in Parkinson's disease, this latency is much more. And we have seen that with probiotics, there is shortening of this more towards the normal. Along with it, we have done the FNIRS studies, which correlates very nicely with fMRI and gives us a much better temporal resolution, and some memory tests, also tests for anxiety and depression scores, because in Parkinson's disease, it is not only cognitive symptoms or motor functions, it is also the emotional accompaniments which go with it, including depression and loss of functionality, so to say. Next, please. Yeah, so these are some results. And next one. Yeah, so these are some protocols which we do in the human subjects. Next, please. So these are the parameters. So simultaneously, we are running in the human subjects a clinical trial and also a blinded study on the rat model. And so that we can then compare our results and have the whole gamut of translational approach to evidence, which I think is the right thing. So next, please. Yes. I think there's an animation here. Okay. Yeah, so this is the design of the study. So we are doing immunohistochemistry, we are doing patch clamp with biophysics, and there are a number of 15 investigators along with it. This was a DST project and also then a DBT project. So next, please. I think we can move further. Yeah, this should be over. Yeah. So that's okay. We don't need more details than that. Yeah. Thank you so much. Sir, I hope it answers some of the questions. So in normal aging also, like Dr. Mohan mentioned about primordial intervention, we in physiology go slightly earlier than even preventive health. We do what we call promotive health. Along with it, we are doing this in controls also across the age groups. But because the studies right now are blinded, so maybe 2-3 months, we will do unblinding and publish it in a proper way. So some publications are already there. They can be accessed on Google.
Thank you very much, Pat. I think you give her a big hand for all the good work they're doing in the AIMS. And this really offers hope for the aging population in India, that many of these neurodegenerative diseases we could manage at least reasonably, make them functional. And that itself will be a great achievement. Continuing on aging. Yes, you also mentioned. Yes.
And we are also working on cerebral palsy as well as ADHD, neurodevelopmental disorders, but those results are not here with us right now. So,
Sure. Now we'll continue on aging. We have Professor Seema Puri, Professor from Institute of Home Economics, University of Delhi. She would be speaking on, is there evidence to suggest that probiotic actually increase longevity? I actually wouldn't welcome it, in fact. Like the increased longevity, then this neurodegenerative problem has to be managed, isn't it? So it's a kind
You
of
double dose. You double dose, you'll prevent both.
Go ahead, Dr. Seema. That's okay.
So I think my life has been made easier because a lot of the discussions and deliberations we've had in the last few hours have been on different dimensions of aging. And there's been hardcore evidence coming in regarding the evidence on different dimensions of aging. I would just like to mention here that as people have said earlier, sorry, I think this is, yeah. So with age, there is a decline in the diversity of microbiota. And basically, we've seen that there's a lot of evidence coming in where the metabolic processes which affect cognition, body composition, and immune function by the gut microbiome have been established. And if you're able to modulate these processes, it would contribute to healthy aging. One aspect that has been talked about by Professor Childs in her earlier presentation was inflammaging. And we do know that with age, the gut microbiota, the intestinal permeability changes, and therefore certain microorganisms or their metabolites can get into circulation. And this then generates an inflammatory response, which is a low-grade inflammation, and this is called inflammaging. And this in turn is responsible for several diseases now, right from cardiovascular disease, cachexia, frailty, cancer, and as ma'am just mentioned, they're exploring the neurological disorders also. So basically, it's the dysbiosis in the intestines along with this inflammation, which may be the reason for the aging process as well as the superimposed NCDs which may be happening. So frailty is something I would like to talk about. And we don't know that as we grow older, the production of the short-chain fatty acids in older adults decreases. And it is in some way related to insulin resistance, intramuscular fat accumulation, and decreased muscle function. And therefore, gut dysbiosis could be related to sarcopenic obesity. We also know that sarcopenia and frailty related to malnutrition may also be attributed to the gut microbiota, because the metabolites, the microbiota metabolites, they modulate satiety and appetite. And if these are affected, it could lead to malnutrition in the elderly. For many of the young people here, we know that malnutrition in elderly cuts across all income groups, it's not due to poverty, and it's due to many socio-psychological and maybe now physiological factors. And therefore, the gut microbiota modifications, which are age-related, may be capable of triggering off the frailty syndrome. We also find that short-chain fatty acids influence bone metabolism, either by regulating the inflammation or the osteoclast activation, or they influence the calcium and magnesium absorption. Immunity, I won't go into details. We've had a very excellent presentation earlier on immunity. And we do know that with age, there is immunosenescence, where it's a low-grade inflammatory state, intestinal dysbiosis, oxidative stress. And this influences the gut microbiota and also could influence the functional ability of the elderly. Neurological health, ma'am is the expert here. But just to say that the gut-brain axis, which everybody's talking about today, we know is bidirectional. So changes in the gut microbiota can trigger cognitive impairment. But we also see that neurodegenerative diseases with an accumulation of beta amyloid peptides can lead to generalized inflammation and change the conformations of the gut microbiota. So it's a two-way relationship. There are negative associations between the microbiota and various functions like memory, decision-making speed, fluency in adults, older adults. And as ma'am has said, there's some work going on in Parkinson's, Alzheimer's, and how probiotics may be helpful. One study has identified 4 prior probiotic strains which were isolated from centenarians. And these were said to protect against neuroinflammation. But this is a study in animals, and a lot more evidence is needed from human studies. So just to conclude, I'd just like to flag it off that the institute in Guwahati has discovered a next generation probiotic bacteria from a dairy product, which they say will help in healthy aging. And it's generally the bifidobacterium and the lactobacilli genera, which are the most studied in clinical trials. But again, which probiotic may have a particular application in elderly population, how much and what to give, really needs to be substantiated with rigorous scientific trials. Okay, so that's just what I want to say here. Regarding longevity, I would just like to emphasize here that a recent study in China has isolated Bifidobacterium adolescentis from the gut of centenarians. And they also found that when they introduced this bacteria in the mice model, they found it reversed osteoporosis, neurodegeneration, and other age-related conditions. And therefore, maybe this bacteria could would have a potential in slowing down the aging process. So again, like I said, a lot more needs to be done to substantiate the role of probiotics in slowing down aging. Thank you.
Thank you, Dr. Seema. In fact, if you go back into the history of the whole science of probiotic, it actually started with the observation that people who ate fermented products in one European country actually lived longer than anywhere else, led Metchnikoff and subsequent studies. So we come back one full circle back to trying to look for ways and means of preventing the problems related to aging. Now we come to the last of the panelists, who is Professor Ajay Balla, Director and Head Department of Gastroenterology and Hepatology from the Fortis Hospital. Professor Balla, I have 2 questions and very important ones. One is, can probiotic help in alcoholic liver disease as well as non-alcoholic fatty liver disease?
I think alcohol is a popular drink. You know, there was a time when people used to ask, what's your favorite drink? And people had aerated drinks or juices. As a kid, I used to write water as my favorite drink, but no more. Now, in the era of single malts and every other thing, alcohol surprisingly affects only 20% of people who take alcohol regularly. In the sense, only 20% population actually gets liver disease. 80% of people may get fatty liver and they go scot-free. So what has been studied mostly is that this 20% is the one who has problems. Maybe it's a problem with the gut microbiome. Maybe it's a problem with their metabolism, which is genetic. It's probably familial. So there is definitely a role of probiotics, which they talk about, that if you were to look at it, if you change the microbiome, probably the tolerance to alcohol and their progression to what would we call as end-stage liver disease could be prevented. So there is role, but there is not enough data, which is there. But you look at it, I'm at a center, which is a liver disease center, where they are using FMT as part of therapy for alcoholic hepatitis. Alcoholic hepatitis is a condition where in a pre-existing liver disease due to alcohol, suddenly there is deterioration of the liver function. The person has features similar to a viral hepatitis. There's very high enzymes, liver enzymes, and they go into what is called as acute liver failure.
So these are patients where a trial is being done and there are positive results which are coming out. There is some hope because otherwise alcoholic hepatitis has a mortality of close to 33%. And these are patients who may or may not even be ready for a transplant. So you have to offer them something. So FMT is a model which they have used here. Non-alcoholic fatty liver, the other part, of course, metabolic syndrome, diabetes, non-alcoholic fatty liver are all together. They're all grouped as one. And there is enough data. In fact, day before or yesterday also, we started with that old concept of micro-obesity in rats, how the experiment was done that when a thin rat was put into a fat rat group, he became fat because rats are coprophagic, they eat their own feces. That's how it happened. So there is definite evidence of metabolic syndrome improving with probiotics. The entire microgenome can change and that would help. So I hope that answers. Yeah.
Thank you. Thank you very much, sir. And now it's open to audience and you can ask the panelists any question. You can either direct it to any specific panelist or you can ask a question in general and one of the panelists will answer. Anyone? Yes, Professor Ganguly, sir. Sir, can you use a mic, please? Can someone give him a mic?
Dr. Balla, when I go for a party, I take 2 bottles of Yakult, and it prevents me because otherwise I get very severe gastritis, acidity, and I have seen with the regularity that I don't get it if I take it before. So there has been other drugs also, hepatoprotectives, like Picorazza Corora, the Picrolive, which CDRI did. That was also a pre-party drug. So could somebody do research for that, whether Yakult could be a pre-party?
No, I can answer that, sir. I'll answer that. The good part or the bad part about alcohol is the absorption starts from the oral cavity itself. And by the time it reaches the second part of duodenum, 90% of alcohol is absorbed. So if you want to slow down your absorption and cause less damages, that before a party, have a burnt toast and a slice of cheese. It will delay the absorption. Charcoal will delay the absorption, so does the protein. So that will help you more. But I don't know if Yakult will help you immediately. Long term, it will definitely help you. And sir, your microgenome, I would like to study that because I'd like to acquire the same.
Sir, I hope this information doesn't go out because liquor stores will also keep Yakult in one of the shelves.
Oh, yeah.
Any other question, please? Yes, gentlemen there, the back. Oh, no, you can go ahead and then later on go to him. Yeah, yes, ma'am.
Dr. Mohan,
Yes.
I want to know if we take probiotics regularly, would we keep the endocrinologist away?
Well, I hope that becomes possible because then we can change our specialty. And that's what I'm waiting for. I'll get rid of all the people with diabetes.
No, but you're talking so much of reversal of diabetes and pre-diabetes, but really on a serious note, if we take it regularly, can we stop the pre-diabetics going into a diabetic stage?
So as I showed you, the pre-diabetes data is not strong. So we need more data. In diabetes, it seems to work better. Poorly controlled diabetes, it seems to work better. And older people with diabetes seems to work better. But in those in type 1 and pre-diabetes, the data is as of now poor. But obviously more studies need to be done. I didn't see any Indian data. So I've been asking, Nirja and others to support some studies in India. So let's collect our own data. We are doing some studies. I'm looking at a large study on sweeteners, because that's something which is supposed to, and we'll be having another meeting soon with Dr. Shikaran. So the sweeteners are supposed to, completely destroy the gut microbiota. That doesn't seem to be true, actually, because in the studies which I have done, not yet ready for publication, but we've got initial results. And then there are other situations where it should be. If I can make one comment, because people from dairy are also here. There is a common myth that dairy is very bad for diabetes, for metabolic syndrome. And I know that particularly in western part of India, from Maharashtra, from Gujarat and so on, particularly in Maharashtra, There are a lot of people, including dietitians, who say that you simply stop milk and you
Yeah,
simply
why?
stop dairy and diabetes will go away. We are finding the exact opposite, actually. It started first with cross-sectional studies, which we didn't believe ourselves, which just when you're looking at it, we found that dairy was protective, milk was protective. So we said this may be some other confounder, maybe they are healthier or something. Then we did prospective study, prospective longitudinal studies. And the pure study was done on 150,000 people followed for 15 years in 23 countries. Now, this cannot be, and this is longitudinal data, incident diabetes. And what we found was that milk was protective, yogurt was strongly protective, and total dairy was also protective. Things like butter and so on were not protective, but they didn't have any bad effects as well. So I would definitely encourage the use of dairy in people with, and not only prevented diabetes, but also incident hypertension and metabolic syndrome. Now, these are 15-year follow-up studies. We have not done randomized clinical trials because we don't know how long to continue them. But the evidence that we have now is that certainly dairy is not harmful for people with diabetes. If at all, I would say it is protective and it should be taken by people. And I think that message, it's not definitely a probiotic as such, but in the context of this discussion and the fact that a lot of people from the dairy institutes are here, I wanted to bring this message loud and clear.
So can we take it at bedtime too? Late nights dairy, yeah? Yeah, it has a
Each
certain...
body is different and there's no one size fits all. But during the day, any time, I think you should include, some people say when they take that, they don't feel good if they take it at night. That's all individual. But I think inclusion of dairy, total dairy, milk, each component, particularly yogurt, is coming out very, very strongly protective. We have another paper from our own group, which is now under publication. A systematic review also we have done, which is also showing the same thing. So I think it is definitely protective. No doubt.
Actually, Dr. Mohan.
Yes, sir.
Dr. Gopinathan, who was a neurosurgeon, who was a cardiovascular surgeon, was the teacher of Dr. Venugopal.
Yeah.
He came to me one day and said that I want to do research. I was doing apolipoprotein assays at that time. Nobody did it in India at that time. So he said that ghee is a very good thing to take, as it was taken in ancient India.
Yeah.
His theory was that the LDL in this ghee has got a core of vitamin E, and that's a very strong antioxidant. So it prevents lipid oxidation. Lipid oxidation and scavenger receptors are very strong indicators, very strong, but they actually promote the foam cells and other lipid cells. So he did a study, and after he retired as a cardiac surgeon, and it was such a seminal study that he showed that the ghee, as it is made in India, is very protective. So dairy, there are peptides, as you have said, in the milk and others, which calm you down, which induces sleep. There's so many things in the milk which are beneficial. So very thank you for bringing this out, that dairy is not a banned product. And Seemapuri, there is a girl who's sitting here. She did this Guwahati study. Her name is Ayushi Purohit. She was here in the morning. I don't know whether she is here. She has got this patented probiotic strain, which actually helps in the age, will help in the aging. So this is something, and I was also very excited. I thought that she deserved a very good prize. There's a girl from the ILS who did a tribal probiotic, identified tribal probiotic strain, which could actually replace salmonella. I've never seen this kind of a data before. Was very, very impressed by that particular thing. So I will request Nirja to give her the full prize, not a consolation prize. That was a very good study, and We need such studies of identifying unique one. That this centenarian idea was very good. That why not look at what kind of
Thank you, sir. Actually, Mohan, there's a publication. You said there may be around 400 and odd bioactive molecules in the milk itself.
Yes.
Many of them are closely interacting with the gut hormones.
Yeah.
Therefore, that is how the milk is probably functioning. And we need to do a lot of research in dairy.
Sir, I would appeal to you, sir, because, whenever I talk about dairy, I'm looked down upon because, this vegan group is so active, and that's for different reasons. See, they are mixing up cruelty to animals with taking milk and so on. They're two different issues, isn't it? Treat your animals properly. We're not saying go and harm the animals. But that doesn't mean that, vegan is superior to something else, and dairy is bad, and milk is bad, and yogurt is bad, and everything is bad. There are a lot of people saying you just stop there and your diabetes will go away. It's far from the truth, sir. It's not at all true.
Yeah, you go ahead, sir. You've been waiting for a long time. And we'll take just another question if time permits, and then we'll conclude this session. Go ahead, please.
Question for Dr. Mohan. So in your probiotic studies, did you see any changes in blood insulin when you conducted them?
So I showed I have not actually done studies on probiotic. Let me clarify. These are all meta-analysis and systematic reviews which we have done. I'm not sure about Indian studies. What we are looking at in our own series is we're looking at acarbose in one study and seeing whether it favorably alters the gut microbiota. Metformin separately we are studying. Then we're looking at sweeteners and looking at that. They're all independent studies, which of course we have reported on the gut microbiota in diabetics, pre-diabetics, and normals in Indians and Danish, and that's all been published, in fairly very good journals. But in one study which I showed in the meta-analysis, it did reduce the insulin levels. So it did reduce hyperinsulinemia. So insulin resistance obviously being cut down. And I think more studies need to be done. In our country, particularly, it needs to be done because the high carb load that we take, how does probiotics and dairy in the background of a fairly high carbohydrate, how does it work? It's worth doing, and a nice RCT.
Anyone has another question?
Yeah.
Right at the back.
My question is from Dr. Seema. Did you use some other model organisms like more age appropriate, like nematodes, to check bifidobacterium or their metabolites, whether it increases health span or not? Or does it trigger like insulin pathway to extend health span or some other pathway?
This is just work that I have reported. I have not worked on this myself. These are just this is just material that I have compiled from reviews, and I have not worked on these myself. Okay, so I can give you the references of those papers, and after the talk, and you can surely go through them.
Thank you. I thank all the panelists for their excellent, overview. And I'm sorry that we don't have sufficient time. Otherwise, we could go on with extremely interesting discussion. And a lot of new areas have come up in the respective fields. And also thank the audience for the active participation. And we look forward to more discussion in the next meeting. Thank you. And thank you for giving me this opportunity. Thank you, Mohan, for holding on.
My pleasure, sir. I'm sorry I couldn't be there with you, and enjoy the hospitality next time.
Thank you to our venerated panelists and our moderator for curating the entire session so very diligently. Can we have a round of applause for them, please? I would now request the moderator, Sir Dr. B. Sasikaran, sir, to kindly step forward and felicitate all our panelists on board one by one. Starting with Dr. Nomita Ma'am, please come. Yes, so we have Dr. Nomita Chandiok, ma'am, here. Thank you so much, ma'am, for your eminent presence here today. Please accept this token of respect, this token of gratitude from our end. Next, I would like to call upon Dr. KP Kocher. Thank you so much, ma'am, for your humble presence here today. Please accept this token of respect from our end. Thank you so much. Next, I would like to call upon Professor Seema Puriji. Once again, thank you so much, ma'am, for coming in here today. A delight to have you here with us. Thank you. Last but not the least, I would request Professor Ajay Bhala sir to please step forward and accept this token of respect from our end. Thank you so much, sir, for coming in here today. A delight to have you. Also, I would request Dr. Sasikaran sir to please stay back and stay put with us as I welcome Dr. Santosh Pasi sir to please join us here. So we have Dr. Santosh coming in here and delivering the memento, token of appreciation to Dr. Sasikaran now. Can we welcome the lady with a round of applause, please, everybody? Hands up in the air. Thank you, ma'am. Thank you to our moderator here. Thank you so much, sir, for curating the entire panel with such accuracy and diligence. Thank you so much.
Thank you.
Thank you. Once again, I would request our panelists to kindly take seats. Thank you once again. Yes, please. So one group photo. Yes, ma'am, if you would want. Dr. Santosh, if you would like to join in, please do come. Please, thank you. Thank you once again to the dignitaries here on stage. Kindly take your seats downstairs. Thank you. As all good things come to an end, today's event is also coming to its closure. For vote of thanks, I would now be requesting the humble presence of Dr. S.M. Shastri sir to kindly come here and say a few words and conclude this event, this symposium effectively. Let's welcome Dr. S.M. Shastri sir with a round of applause, please.
Yeah. I think now the term gut feeling is making more sense after the conference. Yeah. On behalf of the Gut Microbiota and Probiotic Science Foundation India and ILPS, I am privileged and honored to propose the vote of thanks. Firstly, I would like to thank Professor Ganguly sir for giving us the honor of organizing this symposium at our institute, ILPS. I would like to thank our very own director, Professor Sarin, for his interest in this area and co-hosting the symposium. I would like to thank the foundation members and support from all the senior members from Japan, France, and India, Mr. Takeshi Yoshimoto, Dr. Junji Fujimoto, Mr. Eiji Amano, Mr. Tomoyuki Iwama for their support throughout. And we would like to thank all the faculty, national and international, who shared their knowledge and insights, all the chairpersons, panel moderators, and panel members for making the symposium successful. We are humbled by the number of delegates and full house on both the days and also the pre-conference FMT and both offline and online. We would like to thank all the young investigators for all the wonderful work and their enthusiasm in sending and presenting their work. We would like to thank Ms. Suchita Gera and Mr. Ashish from JP Publications for helping the editors, Professor Ganguly sir, Professor
Sasikaran, Dr. Lahiri, and Dr. Nirja in bringing out the book on gut microbiome and probiotics. And thank you to the Mr. Rajiv Pandey, Ms. Ragini, and Vishal for the wonderful effort and all the support. And my special thanks to the ILBS team, IT team, especially Gaurav and his team, the administration team, Colonel Ram, and the ILBS hospitality team, Anurag, for providing the needed support. And my special thanks to Preeti, Anushka, Komal, Smriti, Saili, and Bhagyashree for all their help and support. This symposium would never have happened without your unstinting support. We all thank you all for coming here and for on the weekend and spending your precious time and to make the event very successful. We hope you enjoyed equally. I'm happy to be and privileged to be part of this. Thank you, sir. And I'm sure we'll all meet again next year with the 14th symposium, and definitely we'll have a better time next time as well. Thank you, sir. And yeah, may I request for the anthem? Okay.
One more thing is there. Thank you so much, sir, for the vote of thanks. I would now be requesting Dr. Nija, ma'am, to please do the honors of coming here, joining us here on stage, and presenting the token of appreciation and memento to Dr. S.M. Shastri, sir, as well. Thank you, Dr. Shastri, for your willing support, guidance, for organizing this wonderful, wonderful event. Please take center stage. Please accept this token of respect, this token of gratitude from the entire team for your immense hard work for the past couple of days to bring this event to a success. As all good things come to an end, today's symposium is also coming to its end, and we had 2 really fruitful days of sharing knowledge with each other. As it said, successful people make decisions based on where they are going in life. So always try to envision and entice yourself and push yourselves to reach your utmost limits. On the note of positivity, I would request you all to kindly stand up for the national anthem. Thank you to all our guests and dignitaries here today. We would now be proceeding forward for the lunch hour. The lunch is being served. Kindly don't forget to fill in the feedback forms online and submit them online. Thank you so much for your humble presence here. Kindly move forward for the networking lunch now. Thank you so much.